Everyone. I would like everybody to kindly settle down as we are just going to begin. We all welcome you to the final day of Immunocon 2024. This morning, we have a series of award lectures that recognize some of the best teachers and budding researchers in immunology in the country. We will listen to talks featuring some of these outstanding works of teaching and research in immunology. I request Professor Santosh Kar to chair the Polyphenolics IIS Best Immunology Teacher Award 2024 and introduce this year's awardee. I request Professor Santosh Kar to introduce this year's awardee to Professor Bishwadeep Bishai from University of Calcutta.
Actually, I did not want to do that, but anyway requested, so I'll talk about a minute. Okay. Good morning to everyone. What a wonderful meeting it has been. The past 3 days has been so wonderful. And today, if you sit down and look at it, I will not take more than 3 minutes. When you look at it, you realize behind all this, there are teachers who taught immunology. And that relates to the research that was done. Please let me tell you very honestly that without a teacher, research motivation will not come. Education you can give, but skilling the people can be done by a teacher. So we decided that we will recognize teachers. Last year, we awarded 3 teachers, excellent teachers from the country. We solicited application. Polyphenolics was involved because Polyphenolics financed it. Indian Immunology Society made a list, and finally we decided and we awarded. This year, unfortunately, we got only 3 applications, and that also very late. And the way to evaluate a teacher, we are still grouping with ideas by looking at how many PhD students he has guided or how many publications he has done is not the way to evaluate a teacher. The only way to evaluate a teacher is to talk to the students of the teacher. Talk to them directly, which I did. And here before us today, Biswadev will come. I will request him to come to the dais from Calcutta University. Biswadev Bisoi, who is teaching for last 27 years immunology, microbiology to a group of students. And he gave me names of 5 of his students. 2 of them are abroad and 3 of them in India. And I talked to all of them. Only after that, we decided, the committee appointed by Indian Immunology Society, Professor Sadhana Sarma from Delhi University, recommended him. And then my committee, consisting of Sarad Dalai, myself, and Ajit Sodhi, again went through it and recommended. And finally, Biswadev Bisoi was selected. So his background is that he has created, I think, how many PhD students you have, but at least he has enucleated. His students are now working in Calcutta University, working in Silchar University, working in all over the country. And that is the measure of the success of a teacher. It is not that how many papers he has published, how many places he has nucleated by his students, and where immunology research is going on. The emphasis now is going to shift. Our emphasis of selecting people and giving them recognition on a national platform is going to shift to young people now. We had restricted this year to 50 years plus, but we realized that that was also a mistake. We should make it flexible. And since I said I will only speak for 3 minutes, if you permit me another half a minute, this has been a very emotional affair for us. I have taken the blessings of Professor Talwar. Today morning also I took his blessings. He is all with us. And just imagine Professor Talwar sitting in AIIMS, initiated the immunology group consisting of very few people. I was not one of them. I was not back in India at that time. But when I came back and joined JNU, I also became a part of that. And today we have 1,600 members. 800, almost 900 people voted in the election process. And Dipankar tells me, of course I have not counted, about 300 posters. How that was possible? Because of one person, and he's still sitting here at 98. I am turning to be 80 now in another 2-3 months, and I wish that I survive till 98, so that I will also continue to promote immunology. With that much of introduction, next year it is going to be held in Prayagraj, IMMUNOCon. We'll initiate the process of selection. Professor Talwar himself had suggested to me in the 49th IMMUNOCon that Santosh, don't ask for application, you select. You do not, because a teacher will not apply. Teacher doesn't care about recognition. Students are his recognition. When a student goes out of his lab and does something good, that is the recognition. He will not apply. So we need to select. And that process should be impartial, very perfect, transparent. And that we are trying to evolve. Any suggestion by any one of us, any of you, can be transmitted to EC, IIS EC, and then we will implement it in selecting. I understand that we need to also recognize women scientists, women teachers. We had recognized one from AIMS last year. This year, unfortunately, one lady applied, but she did not respond, so we could not include her in the selection process. But next year onwards, I promise you that we will try to include ladies, because they're more reluctant than men. Men are a little aggressive. So with that word, because we have very limited time, I will now request Vishwadev to come on stage and say a few words if he wants to say anything.
So with all your blessings, I am here today, and it's a great honor for me, and I feel proud to be here on the Immunocon platform. Actually, I was in the passion of immunology from my previous gurus, like Samal Roy at IICB during my PhD tenure, Bhaskar Saha. So they actually taught me initial immunology and basic immunology things. And after that, they also advised that you must attend the Immunocon conference so that you can learn how advanced immunology is going on all over the country. So I started to attend the Immunocon and also learn a lot of things. And this achievement is all for my inspirations also for future endeavor. And I dedicated this achievement to all of my students. I have completed 22 PhD students so far, and right now 5 students are doing PhD. But you see, in the university level, there is so much constraint without any big grant, and other instrumentation facilities are very few in West Bengal or other universities also. So I actually requested the Immunological Society team. So MSc in immunology can be initiated if you approve, and then you can transfer it to the UGC. So MSc immunology can be started in several universities. That is my proposal to the Immunology Society team. And another thing, you must promote the teachers who are working in all over the country, in the universities at least, because you see the participants from the university level is very less in the Immunocon. So far, my experience goes on. Okay, so thank you very much to the Immunocon Society and all of your blessings, all the professors and my students, my beloved friends. So I am here. Thank you very much. Okay.
Thank you, Mr. Dev. As a recognition of your contribution to teaching and creating centers in this country, I have talked to them and I was extremely pleased. We give you this hall. It is a recognition of honor. And then this is a gold medal, real gold medal, 22 karat gold medal. I sat there, I got it made. It is not those big medals which I have also got few. I don't like to get a gold medal which is coated bronze with gold. This is pure 22 karat gold. Government has now imposed restrictions. Prime Minister's office telephoned me that you cannot give more than 50,000 rupees worth of award. So this is honor of it. Give it to your wife, ask her to keep it safely for the inspiration. And this is a check, 30,000 rupees cash. He can encash it. And this money he can use for taking his family out for a dinner. Do whatever he likes. This is yours, Vishwade, and this is the certificate.
And then the certificate from the Indian Immunology.
So in the present, our president could not come because he was involved in something else. He's tired, but what a wonderful thing Dipankar has done. I would release it till my life, as long as I live. I will release it because of the fact that younger people participated fully well. So please do come and participate in the Prayagraj. And the next one we are going to Srinagar. 26th we are going to Srinagar. 25th is going to be in Prayagraj. And there will be more hours and hours for younger people now. We will now recognize younger people from different regions, not from one place. Different regions of India. In the EC, yesterday, day before yesterday, we discussed, and already Chennai, Ahmedabad, Bhubaneswar, and probably also in Banaras, or wherever is Uma going to be locating herself, we will be activating the immunology activity, student-centric activity, so that students will come forward and we will get an opportunity to select students from there who are enthusiastic and promote them so that they become teachers and then they come. Thank you very much. And thank you, Dipankar, for all your hard work. I've seen you have been sitting through, just like Dr. Talwar sitting through the whole day. You have been sitting there through, and this is my inspiration. Watching Professor Talwar has been my inspiration from the very beginning. I was telling him today morning that, sir, you remember when you started in AIMS. I was not there, but as soon as I came back, I joined there. So thank you very much for this.
Thank you. Thank you.
Go ahead.
Our sincere thanks to Professor Santosh Kar and heartfelt congratulations to Professor Bishwadev Bishai. Moving on, we have two GP Talwar talks. I would like to request the chairs of this session, Dr. Kaustav Nayak, Secretary IIS, and Dr. Gaurav Gupta, Joint Secretary IIS, to kindly chair this session.
Yeah. So, welcome everyone for the most awaited GP Talwar Young Scientist. This is the session which most of us always wait for, energetic presentations. So this year we have two presentations, and these presentations are for 8 minutes plus 2 minutes of discussion, and this is a competition. So these two candidates will be evaluated for the presentation on basic and clinical relevance, if any translational relevance of the work, scientific content and presentation skills, if the work has been done in India or not, time management, and then the question and answer session. So after 7 minutes, Dr. Bala will tell them that 1 more minute is remaining, and after 8 minutes, they'll have to stop. The floor will be open for questions by the judges first, and then by the audience.
Yeah, please stick to the time.
Another, if I can suggest, that once the resolution for 8 minutes is over, it is first priority to be asking questions of the judges. If there is time, then the questions will come. Otherwise, we cannot understand any thought.
Done. Yeah, so let me call Rashmi Gupta to make her presentation.
So, good morning, everyone. I am Rashmi Gupta. So, title of my presentation is Dendritic Cell-Mediated Port Vaccine Approach, a Potential Immunotherapeutic Target for Epithelial Ovarian Cancer. So, title of my study is Epigenetic Potentiation of Dendritic Cell-Mediated Port Vaccination as a Promising Immunotherapeutic Strategy for Human Ovarian Cancer. As ovarian cancer is the most lethal gynecologic malignancy and is the third most leading cause of the ovarian cancer death, the high-grade serous carcinoma is the most common subtype of the ovarian cancer and having a mutation of the P53 and a loss of heterozygosity that will make in India, that is also the third most leading cause of the ovarian cancer death. And in metropolitan cities, the number of the ovarian cancer is raised by year. And the major problem of the ovarian cancer is the multiple relapse. That is because of the late stage of the detection, and also the lack of the early biomarkers. So that will make a difficulty to treat the ovarian cancer. So the era is moving toward the cancer immunotherapy. In cancer immunotherapy, the patient's own immune system will activate or engineer to kill the tumor cells. So these are the immunotherapeutic approach which are used for the ovarian cancer, used for the cancer immunotherapy. So recently the CAR T cell therapy has been used for the lymphoma patient, and also in this study we are using a cancer vaccine therapy for the ovarian cancer. So, as many cancer cells expressing a protein known as cancer antigen or cancer testis antigen. These cancer testis antigen having a limited expression in the germ cells but overexpressed in the cancer cell. That will make a immunogenic aspect for the tumor vaccine, and also CT antigen having a regulation with the epigenetically. So these are the CTA-based clinical trial which are for the which are for the cancer research, and like MAGE, GAGE, and the NBIESO1. There is a newly identified CTA family member is PORT, which is also a CTA family member having a 14 different paralogs which are dispersed on the 7 different chromosomes, also having a restricted expression. So in this study, we are trying to check the antigenicity of the PORT protein, whether it is antigenic. If it is antigenic, then it can be activate the T cell of the ovarian cancer patient. So for that, first of all, we have checked the antigenicity of the PORT protein. Out of the 14 members, the 7 members having an antigenic capacity. Then we have predict the T cell epitope with the help of different server, and we found that these T cell having a binding affinity of the HLA-A2 and HLA-24 and HLA-A3 population in Indian as well as the multi-state countries. Then we have predict the 3D structure of the T cell epitope for the HLA-A2 and HLA-24 and HLA-A3. Then we check the interaction with these HLA with the T cell epitope, and we found that after the molecular docking, the port peptide is binding in the binding group of the HLA molecule. And also we have checked the stability of breach complex with the help of the molecular dynamic simulation in 50 nanosecond run. In 50 nanosecond run, the port peptide is binding the binding group, and the RMSD and RMSF is lower than the 2 angstrom, will indicate that the most port protein is stable in the physiological condition. So after checking the antigenicity and stability of these complex, we have tried to check the antigenicity and T cell activation with the help of these peptides. We isolate the ovarian cancer patient blood sample in AIMS IRCH board, and then we have isolated the PBMCs from these patients and culture these PBMC to mature dendritic cell. And we check the phenotype of the dendritic cell with the help of the bright field SEM and the confocal microscope. Then we have also checked the phenotype of these dendritic cell with the help of flow cytometry. And we found that the co-stimulating marker CD8086 is also expressed in these dendritic cell. And after checking the phenotype and the morphology of dendritic cell, we have paused the port peptide, which is tagged with the FITC. Then we check the peptide uptake with dendritic cell. So we found that the FITC signal in the bright field in the fluorescent microscope and confocal microscope indicate that port peptide is engulfed by the dendritic cell. Then we have checked that whether this peptide activate T cell or not. So for that, we have isolated the CD8 positive T cell with the same ovarian cancer patient and check the T cell activation. Here we have using a control peptide which is not antigenic and the port peptide which is antigenic. Then we check the CD8 positive T cell activation, and we found that the CD8 positive T cell activation is high in the port peptide condition. Then we have also checked the interferon. We have also checked the cytokine level. That is the dendritic cell mediated T cell cytokine that is high, that is expressed in the port peptide condition, will indicate that the T cell activation is there. After T cell activation, we have checked the cell viability of the ovarian cancer cell line. So for that, we have using a co-culture model in 24 and 48 hours. We have co-cultured the dendritic cell loaded T cell with ovarian cancer cell line and check the cell viability. After 48 hours, the cell viability is decreased, and also clip caspase level is also high, and apoptosis is also high in the port peptide pulse dendritic cell in port peptide pulse and ovarian cancer cell line. Then we have also try to check, as we know that the CTA expression is regulated by epigenetically. So we check that whether the epigenetic play a role for the T cell activation and the immunity. So as port is also belong to the CTA family, and port family having the two type of the methylation. It's like hypo and hypermethylation, and port family having a expression in the hypomethylated condition. So here we have hypothesized that if ovarian cancer cell line is treated with the epigenetic modulatory drug, that will enhance the CTA expression. Then enhance CTA expression.
1 minute more.
Enhanced CTA expression will activate the T cell and cytotoxicity increase. So for that, first of all, we have checked the expression of the port in the different ovarian cancer, different port family. So ovarian port family expression is high in the tumor condition, and also in the cell line, the port family expression is high. Then we have co-cultured the port peptide pulse T cell with the epigenetic modulatory drug treated ovarian cancer cell line, and we found that as compared to the control condition, the cell viability decreased in the port peptide pulse epigenetic modulatory drug. That will indicate that the port peptide will respond better with the epigenetic reprogramming. So the summary of this study is the port family having a robust antigenic potency, and port peptide pulse dendritic cell lead to T cell activation. Also activated T cell increase the cell viability, decrease the cell viability, and increase the apoptosis in the ovarian cancer cell line. And also epigenetic drug treated cell line will activate the T cell cytotoxicity. In the future prospect of this study is the current study will be validated in the ovarian cancer tumor derived organoid model for better implication. And these are my publication and awards proceeding. And I thanks to my collaborator
And I acknowledge the epigenetic lab member in New Delhi, and I also thanks to the IAS to providing this opportunity.
Thank you, Rashmi. Now the floor is open for questions from the judges.
Excellent presentation. Rasmi.
Rasmi,
Yes.
is it Rasmi?
Yes.
Who is asking?
Very good work.
Yes.
With a sorry, somebody else wants to ask?
Yeah, judges will ask first, then from the audience, right?
Yeah, I'm a judge. I will not interfere with that process. The judges should first ask questions. When they finish, then the audience can ask. Hello? Good work.
Thank you.
But there is a problem.
Yes, sir.
You are replacing immunotherapy where antibodies are injected, monoclonal antibodies are injected to prevent tumor growth, tumor cell growth, but you are now trying to stimulate the patient's immune system by using dendritic cells, pulsing them with peptides, and using those dendritic cells as a immunoadjuvant or whatever to promote immune. How will you do it? Because hospitals have to be there, lab has to be there. If you want to do that for each patient, you will have limited number of possible. What is your answer?
Sir, actually, we are using a personalized vaccine. The port protein also having expression in the ovarian cancer patient. And there is the two type of the subtype of the patient is the hypo and hypermethylation. In hypermethylation condition, there is the cold tumor. The activation of the immune cells is low. So the port is having the antigenic potency that will activate the T cell that will kill the tumor cells.
No, you did not understand my question. The cancer patients are living in the villages. They have to be brought to a city where there is a hospital, there is a laboratory, there is an interested person who can culture all these things. How will you achieve that? Do you know the number of cases in India now?
Is more than two per year. Is the more than lakhs. More than lakhs.
Okay, so as I said, presentation is good. What is the prevalence of ovarian cancer in India?
It's the third most leading cause.
Hmm?
Third most. The Globicon 2020
Okay,
data.
out of that, you know the different types. Which is the most fatal?
Is the high-grade serous carcinoma. The more than 70% of the ovarian cancer death rate. The high-grade serous carcinoma.
Okay, you have used facts, right? Any problem that came up while you are standardizing or doing with the facts, because it is comparatively a tricky technique to interpret as well as really to standardize well. Anything where you really got stuck for some time?
Yes, I am stuck with the population of the T cell and also dendritic cell population. The population is very low. Then we have using the growth factor and LPS induction and increase the time of the culture. Firstly, we have using 9 days culture, then we have used 12 days culture, then we got the enough population to co-culture with the T cell. Because we are using two co-culture. First co-culture is dendritic cell with T cell, then T cell with ovarian cancer cell. So we need the number of the cell that is much high, and also the problem we are facing is like we need same patient of the dendritic cell and same patient with T cell. The patient is requiring for the 7 days. Within the 7 days, we need the patient blood sample to isolate the.
Okay, I understand. You can pass it.
Rashmi,
Yes, sir.
you mentioned in your introduction slide there are multiple relapses. Why it is so? Is that anything to bear with the pathology of the disease? Why the multiple relapses are happening?
Because of the there is a mutation which is the P53 mutation, TP53 mutation, and also the heterozygosity. In the late stage of the detection, because of the ovarian cancer is not detected in the stage 2. Also, the patient knows about the ovarian cancer is the stage 3A or 3B. That is the too late. That's why the treatment approach in India or in the world is like surgery and chemotherapy that will not work in the stage 3 of the cancer. So that's why we are approaching the cancer immunotherapy that will activate the tumor cells, that will activate the immune cell, that will kill the patient tumor cells.
Okay, you said that actually A2 is a highly prevalent antigen in the population.
Yes, yes.
Did you check the HLA at least for 4-digit resolution in the patients?
We are checking with the Uma Ganga ma'am. The patient
done or you are planning?
We are planning, or then that patient will have HLA-A24 positive, and in that time we are not using a organoid culture. We are using a ovarian cancer cell line. That is the OPCAR4 with also HLA-A2 positive. That's why we are using OPCAR4 cell line and having a very low post-expression. When we treat with the epigenetic modulator, then enhance the post-expression. And the graft rejection is low because of the peptide is also HLA-A2 positive, and the ovarian cancer cell line is also HLA-A2. For our future study organoid model, we have planned to do with HLA typing.
Now,
Yeah, I think, co-chair has a question.
One quick question to you. Can you name any dendritic cell-based vaccine in clinical practice?
Is like NY-ESO1 with dendritic cell, and also the breast cancer vaccine for the PLOS one paper in 2020.
No. I'm not asking the clinical trials, which is in clinical practice being used for treatment or for the therapeutic purpose.
I have not remembered.
And you mentioned about HLA-A2. What is the frequency, general frequency here in Indian population?
Is more
Rough
than
idea.
is more than 60%.
60%?
50 to 60% in all over world, not the Indian.
It's incorrect. Anyhow, you just check your data.
Yeah, so we do not have time, so now we will not be able to take any questions from the audience. So we'll now let me invite Asha Bharadwaj from Department of Biotechnology, All India Institute of Medical Sciences, New Delhi.
Hello everyone, I'm Asha Bharadwaj and I'm presenting here my work on the role of gut resident Tregs in postmenopausal osteoporotic condition. So I'll start my presentation with a brief introduction on osteoporosis. Osteoporosis is a chronic disease which is affecting more than 500 million people worldwide, and in India 40 to 60% of postmenopausal women suffers from osteoporosis. So there is a need of an effective therapy for osteoporosis, and recently the studies have shown the role of immune system in regulating the bone health, and our lab has coined and established the field immunoporosis, which specifically deals with the role of immune system in regulating osteoporosis. Among all the immune cells, Tregs have a very important role in regulating the bone health by inhibiting osteoclastogenesis, which is highlighted by various studies from our group. And in these studies, we basically delineated the role of Tregs that reside in the bone marrow in regulating the bone health. However, recently studies have shown the role of gut permeability and gut inflammation in regulating the bone health. And for this, we further want to elucidate what is the role of gut resident Tregs in regulating the bone health to link the gut-bone immune axis. And for this, we first developed the postmenopausal osteoporotic mice model. And after 45 days of the model development, we sacrificed the mice and analyzed for various osteoimmune parameters. Our SAM, MATLAB, and microCT data showed the deterioration of the bone microarchitecture in the OVX mice, which is further supported by the histomorphometry and the bone mineral density data. To study the association between the intestine and the bone health, we analyzed various parameters such as colon length, histology of the small intestine and large intestine, and gut permeability of the. And we observed that there are no significant changes in the colon length of the sham and OVX mice, whereas histological damages and increase in the gut permeability is observed in the case of OVX mice. To analyze the Treg population, we analyzed Tregs from various gut tissues, that is small intestine lamina propria, large intestine lamina propria, and mesenteric lymph nodes along with the bone marrow. And we observed that Tregs were significantly decreased in the case of gut tissues along with the bone marrow. And also there is a positive correlation in the population of the Tregs in the gut and bone mineral density. To study how the gut resident Tregs are regulating the bone health, we studied their effect on the bone cells, and for this we first isolated the gut resident Tregs. We isolated the gut resident Tregs from the MLN and cultured them in Tregs polarizing conditions for 5 days, and after that we co-cultured them with the bone marrow cells in the osteoclast and osteoblast prime conditions. And we observed that gut resident Tregs in cell ratio dependent manner significantly inhibited the osteoclastogenesis as indicated by the trap staining, and significantly promote the osteoblastogenesis as indicated by the ALP staining, indicating that these cells, GTregs, have role in bone formation. Gut resident Tregs are further divided into two parts, P Tregs and T Tregs. T Tregs develop in the thymus and then reach the intestine, whereas P Tregs develop in the intestine from the naive T cells under the influence of the microbial antigens and the dietary antigens. So to further comprehend what is the role of these gut resident P Tregs and T Tregs in osteoporosis, we analyzed their population in several gut tissues along with the bone marrow with the help of the NRP1 marker, which is expressed by T Tregs but not P Tregs. And we observed that P Tregs were significantly decreased in the case of gut tissues and the bone marrow, whereas T Tregs were significantly enhanced, indicating that the homeostasis of these two cells is disturbed in the case of postmenopausal osteoporosis. Our earlier studies have shown that probiotics have the tendency to induce the induction of Tregs, and therefore to study whether probiotics can also prevent bone health by inducing the development of gut-resident Tregs, we administered the OVX mice with two probiotics, Lactobacillus rhamnosus and Bifidobacterium longum. And our data demonstrated that along with improving the bone health, these probiotics significantly enhance the population of the gut-resident Tregs, along with restoring the balance of the P Tregs and T Tregs in the gut tissues. To study the mechanism that how probiotics are inducing the development of gut-resident Tregs, we perform targeted metabolomics for the short-chain fatty acid because these probiotics are known to produce short-chain fatty acids. So we perform metabolomics for acetate, propionate, and butyrate, and we observed that in case of OVX mice, SCFS level was significantly decreased, whereas probiotics treatment significantly restored the level of gut-resident of these SCFAs. We further study what is the effect of these SCFAs in the induction of Tregs, and for this we isolated the naive T cells from the gut tissue MLN and cultured them under the influence of the short-chain fatty acids, acetate, propionate, and butyrate. And we observed that short-chain fatty acids significantly enhance the differentiation of the Tregs, and these Tregs, they basically significantly enhance the differentiation of P Tregs, whereas inhibit the differentiation of T Tregs. Butyrate, which is the most potent metabolite, decrease the secretion of the inflammatory cytokines from the P Tregs, further enhancing their immunosuppressive potential. Next, we want to study whether SCFAs prime Tregs SCFAs further enhance the osteoclastogenic potential of the gut-resident Tregs. And for this, we co-culture the SCFAs prime G Tregs with the bone marrow cells in different ratios, and we observed that SCFAs prime G Tregs have more osteoclastogenic potential than the control Tregs. And not only these, SCFAs have the potential of directly inhibiting osteoclastogenesis also, and therefore promote bone resorption, promote bone formation. To further corroborate our mice data with the humans, we recruited IBD patients. We recruited IBD patients because it was difficult to get the colon tissue from the osteoporotic patients, and also IBD patients have a high proximity of getting the osteoporosis. So on the basis of the T-score, we divided the IBD patient into three groups: control IBD patients with osteopenia, and IBD patients with osteoporosis. And we analyzed the Tregs in their colon tissues, and we observed that gut-resident Tregs were significantly decreased in the case of IBD patients with osteopenia and IBD patients with osteoporosis as compared to the controls. And also further, there is imbalance of the P Tregs and T Tregs in the case of IBD patients with osteopenia and osteoporosis as compared to the controls, validating our mice data that in case of osteoporosis, there is decrease in the level of the gut-resident Tregs, and further there is imbalance of the P Tregs and T Tregs. So to summarize, we can say that in case of postmenopausal osteoporosis, there is decrease in the levels of the gut-resident Tregs, and further there is imbalance of the P Tregs and T Tregs. G Tregs have the potential of inhibiting osteoclastogenesis and promoting osteoplastogenesis. However, the treatment of the probiotics in the postmenopausal osteoporotic conditions with the probiotics enhance the level of the gut-resident Tregs in SCFS-dependent manner, which promote the differentiation of the naive T cells into P Tregs. And these SCFAs-induced G Tregs have higher potential of inhibiting osteoclastogenesis and therefore preventing bone resorption. So these are some of my publications, book chapters, and abstracts published in journals. I want to thank my mentor, Dr. Rupesh Kumar Srivastava, lab mates, and our collaborators, all the funding agencies, along with IMMUNOCON 2024 for providing me the platform to present this work.
Thank you. So questions from the judges?
Excellent. Good presentation. What I wanted to just ask you in brief, you have presented all right. Just brief, say what had been your research question? I mean, how you organized the study, with which question to start with?
Ma'am, actually, there are various studies that have shown that gut is modulated in the case of bone health diseases like RA or various diseases. It is shown, and we have studied that probiotics have a potential to preventing bone health. So we want to study whether probiotics are working through gut-resident Tregs, because by enhancing, because gut inflammation is observed in case of osteoporosis. That there is in postmenopausal condition, it is observed that gut permeability is increased. So we want to study whether by using the probiotics or in case using the probiotics, if we are able to enhance the gut resident Tregs or preventing the increase in gut permeability, we can prevent the loss, bone loss in case of postmenopausal women.
What next you want to do? Suppose
Ma'am-
it is left to you.
Ma'am, actually, our clinical trial on probiotics is running on right now. So we are further corroborating our mice studies with the humans.
Okay.
I have a very generalized question. Gut microbiome
Yes, sir.
have been understood to be playing major role in our health. They not only control the environment in the gut, they also have developed an access with the liver, with the brain, with the lungs, and any other organ of the body which are involved. So when you are doing a clinical trial,
Yes, sir.
selecting a probiotic which is going to help in osteoporosis, how are you going to, I'm talking about application, looking at it from a translation point of view. How are you going to monitor its effect on the lungs, its effect on the liver, its effect on the brain?
Okay, so sir, right now in the clinical trial, we are not considering these organs, but yes,
You
this
have to consider, because
Yes,
you
we
are
have to consider these organs.
administering probiotics which are going into the gut and promoting, helping in osteoporosis management. But how do you monitor what is happening in the brain?
Yes, sir.
Because gut is also involved in liver, gut is involved in brain, gut is involved in lungs.
Yes,
So
sir.
you have to you cannot help a patient in bone marrow, osteoporosis, but that person dies because of brain problem. So that is the point that I'm trying to make. And you're talking about clinical trial in a human.
Yes, sir.
So if you are going to develop a clinical trial methodology, you have to also think about your probiotic treatment. How is it not affecting the brain function, the liver function, and the lungs function, while it is improving the osteoporosis?
Yes, sir. We will consider this.
Dr. Asa,
Yes, sir.
in your work, you chose in all the experiments female mice, right?
Yes, sir. Because we are developing.
C57B,
Yes, sir.
L6. Why not male mice?
Sir, because we are developing postmenopausal osteoporotic mice model, so it may make the condition of postmenopausal
Don't you get
osteoporosis.
any inference if you use male mice?
Sir, we can done on mice, but we
You
are
did.
just
You did that.
no, sir. Right up, we are not.
Okay.
Yes, sir. But we can do the same in the male mice for
Even if
male
you do that,
osteoporosis.
can you get any good inference to compare to the postmenopausal model?
Sir, because postmenopausal osteoporosis is very common as compared to male osteoporosis. So we are studying, but right now there are studies that have shown that decrease in the testosterone level can lead to male osteoporosis. So by removing the testes, we are developing this model in the lab to study the male osteoporosis also.
And then the another point, there is a complete opposite trend for this P-Tregs and T-Tregs.
Yes, sir.
What is the crux of that point?
Sir, actually, gut microbiota or gut favors the development of the P-Tregs because P-Tregs are induced from live T cells by the gut microbiota and their metabolites. So the condition in the gut favors the development of the P-Tregs. And we, SCFAs particularly, which are decreased in the case of the OVX mice, these SCFAs have the potential to induce the development of the P-Tregs. So because of the imbalance in the population of the metabolites, there's decrease in the level of the P-Tregs. And because Tregs are, because we are using the marker NRP1, so one is decreased, another one is increasing. So we are saying that the decrease in the deficiency of SCFAs in the OVX mice is leading to the decrease in the frequency of P-Tregs in the OVX conditions.
Yeah.
I have a small comment. You didn't have any access to any pointer or for that matter movement of the mouse?
Yes, ma'am.
Did you have?
Yes, sir.
You didn't show anything.
Okay.
And it was very, very figures.
Okay, ma'am. I thought mouse is a. Yes, ma'am. Yes, ma'am. Yes.
Yeah, nice presentation, Astha. Nice
Yes, sir.
presentation. So, do you think that BREG aberration is also going to affect the IBD in context of osteoporosis, or somehow there is a link between?
Sir, IBD itself is a inflammatory
so-
disorder.
So, what is your take on that?
Yes, sir. That's why, because we are taking IBD patient, control IBD patients, IBD patients with osteoporosis and IBD patients with osteopenia. So we compare within these IBD populations, and we've observed that in case of IBD patients with osteoporosis and osteopenia, there is further decrease in the level of gut-resident Tregs or a Tregs in the colon as compared to the control IBD patients. So if IBD patients are suffering from osteoporosis, their Tregs will further decrease.
Thank you.
Thank you. Thank you, Asha. Thank you, Rashmi. So we'll all have to wait for the results till the valedictory function. So it's over to the organizers.
Next, we have the oral presentations. These talks were selected by the committee based on 359 posters submitted this year. We have 6 talks today, which feature a few outstanding research selected from abstracts that students from different institutions across the country submitted. I request Professor Sandeep Ishwarappa and Professor Utpal Nath from IISc to kindly chair this session.
Good morning. It's my pleasure to start this session of student presentations, one of the most awaited sessions of the day. Okay, there were 360 posters submitted for the Immunocon, and there were more than 20 evaluators. Each abstract was evaluated by at least 2 independent reviewers, anonymous, and they were evaluated for completeness, novelty, and also relevance to immunology. Out of that, 6 were selected for oral presentation. So the first speaker is Babita Negi. She's from ICMR, National Institute for Research in Reproductive and Childhood, Mumbai. She's going to talk about IRGM1 and inflammation during embryo implantation. Babita? So each speaker will have 8 plus 2 minutes. 7th minute, I will remind you. Okay.
Just a minute.
You just use this.
Okay, you can use a pointer.
Thank you so much. May I start? Yes, thank you. Good morning to one and all. Thank you so much to Immunicon for letting me present my work here, which is titled as IRGM1 is required to control inflammation during embryo implantation. This work is being carried out under the guidance of Dr. Deepak Modi at ICMR and IRCH, and it is in collaboration with Dr. Santosh Chauhan from CSIR CCMP. To start with, autoimmunity majorly affects women as compared to men, and these women suffer from high infertility as compared to the healthy women. Now, what leads to this higher rate of infertility is what we are trying to understand here. One molecule named as immunity-related GTPase M plays an important role in maintaining immune homeostasis by regulating different pathways, including inflammatory pathways, interferon pathways, and autophagy. Interestingly, this IRGM molecule has been seen to be a genetic risk factor in several autoimmune disorders, and polymorphism in IRGM has also been found out in women with infertility and autoimmunity. Now, we know that women with autoimmunity suffer from infertility, and this IRGM alteration is also there in autoimmunity women. Now, what is the relation between IRGM and infertility? Is it playing a role in the increase in infertility? Which makes me ask the question that does loss of IRGM lead to infertility? And to look into it, we have formulated the aim to look at the role of IRGM1 in embryo implantation and early pregnancy. So to start with our study, before that, we have used IRGM knockout model for for our study, and towards that we have started checking. We started checking with the fertility profile of the IgM knockout. For this, we have chosen two criteria. One is litter size, and other is time taken to conceive. We could clearly see, compared to wild type, the IgM knockout produced approximately half the litter size, and they took several folds higher to conceive, which tells us that these mice are not completely infertile, but rather subfertile. So, what is leading to this subfertility? Is it an ovulation defect? So, to check that, we looked at the estrous cyclicity for the consecutive three cycles in wild type and IgM knockout. We could see that the pattern of the estrous cycle remains similar in wild type and IgM knockout, telling us that this is not an ovulation defect. So, next question was, is it an implantation defect? So to check that, as implantation is an indispensable process when we talk of initiation of pregnancy. So to look into, we have chosen the histological analysis of wild type day 5 and IgM knockout day 5 uterus. So in wild type, we could observe the very correct formation of implantation cup here, and inside there is an embryo which is aligned to the cup. Surrounding this implantation cup and embryo, there is a proper decidual zone. As compared to that, in IgM knockout day 5 uterus, we could see that there was defective implantation cup formation with the epithelial slugging off at one side, and there was reduced zone of decidual, telling us that it is indeed an implantation defect. Now, what is leading to this implantation defect? So for that, we went ahead with RNA-seq analysis of our wild type day 5 and IgM knockout day 5 mice uterus. And after subjecting it to the RNA-seq analysis, we found out that there were 277 genes in total, which were differentially regulated, of which 82 were downregulated and 195 were upregulated. These genes were then put through the gene set enrichment analysis via using different tools. And in the biological processes, out of these top 20 hits, we could get that the top-notch process altered was response to type 1 interferon along with chemokine production, telling us that there was alteration in interferon response and inflammatory responses. Talking about molecular functions and KEC pathways, here we could see that T cell receptor, NK cell signaling, and via complement coagulation and RIG1 like receptor signaling were affected, in all telling us that there was immune alteration at high level. Now, of these, what is happening when we talk about response to type 1 interferon? Are these genes getting upregulated or downregulated? So we chose a few of the top immunity interferon stimulated genes, and we could see that in the heat map, they were highly upregulated in the knockout samples as compared to wild type. We went ahead with validation of two of the genes, MX1 and ISG15, and we could see that in compared to wild type, IgM knockout expressed it at a very higher rate, several folds higher, telling us that at the time of implantation, there is upregulation of interferon and inflammatory responses in the IgM knockout mice. Now, from the previous study of our labs, we know that this inflammation increase leads to the receptivity failure. To give you an analogy, what is receptivity? Is basically the time where endometrium is conducive enough to accept the embryo because it is a semi-allogenic entity at the end. So we looked at the receptivity markers. A few of the hallmark markers are estrogen and progesterone. So first we looked at ER alpha and ER beta on day 5 in the mouse uterus. So in the wild type, we observed that in the wild type, there was very base-like expression in the stroma, and there was no expression in the epithelial zone. However, in IgM knockout, this ER alpha, ER beta expression was very highly elevated, and we could observe it in stroma as well as in the epithelium. The next marker we looked at is Ki67. Why? Because epithelial marker, estrogen receptor leads to the increase in proliferation. And next we wanted to check that because under the progesterone hormone, this fate of cells needs to shift from proliferation to differentiation. So we wanted to check whether they switch to this fate or not. So in the wild type, we could see that Ki67 was present, but at the distant stromal cells, not at the implantation site or not even in the nearby decidual zone. However, in IgM knockout mice model, this Ki67 was present in the epithelium as well as in the stroma, telling us that these epithelial cells fail to exit the cell cycle and fail to enter the differentiation, maybe leading to the receptivity failure. The next marker on the list was progesterone, and downstream to progesterone was hoxaten and beta-catenin signaling pathway. For the hoxaten, we could observe the baseline expression in wild-type day 5 mouse uterus, in the stroma as well as in the epithelium. However, in an IgM knockout, again, we could see the elevated expression both in stroma and epithelium. Coming to beta-catenin, wild-type D5, there was baseline expression in the stroma and not as much as in the epithelium. However, IgM knockout showed very high expression in the epithelium and glandular epithelium, telling us that again these epithelial cells fail to switch their fate and fail to give the proper receptivity signaling. To summarize till now, IgM knockout mice are subfertile due to implantation defect, and this loss of IgM1 leads to upregulation of interferon response and inflammation at the time of implantation. So this increase is leading to the compromised receptivity and thus subfertility. The next question we wanted to see is that can this subfertility be rescued by blocking interferon response? So we
have 1 more minute.
Yes, sir. So we went ahead to check in the 3 different groups, wild type, IgM knockout, and double knockout. And very beautifully, we got to see that this DKO, double knockout for IgM and interferon alpha, could rescue the phenotype by giving us the same letter size. And the time taken to conceive was also reduced by and was similar to wild type, telling us that now to conclude that high infertility rate, which is seen in women with autoimmunity, could be because of defective IgM1. So to summarize, the semi-allogenic embryo is not accepted by the endometrium if there is an increased interferon response or inflammation. However, if IgM is present, this is regulated and the implantation becomes successful. With that, I would like to acknowledge my PI who has been the constant support and guiding light. And thank you to Dr. Santosh Chauhan and Ramya and Ringu for helping me with all of the work. And thanks to Dr. Sarmishta Shamyal for helping me with RNA-seq analysis. These are my references. Thank you so much.
Thank you, Babita. We have time for a couple of questions. Short questions. Okay.
In a bone-associated problems, I have a very simple question. Did you check the calcitonin levels?
No, sir. We are not looking at it from that point of view yet. Maybe in future we can look.
Okay.
Okay, if there are no more questions, we can move on to the next speaker.
Thank you so much.
Priyakshi Gogoi from Jawaharlal Nehru University. She's going to talk about bacterial membrane vesicles and monocyte recruitment.
The PhD. Yeah. So should I start? A very good morning to everyone. First of all, I would like to thank the committee members for giving me this opportunity to present my poster in this oral poster presentation. So the topic of my poster was Pseudomonas quinolone signal trafficked by bacterial membrane vesicles impair monocyte recruitment and differentiation in alveolar epithelium. So to give you a brief description of the alveolar epithelium and the pulmonary innate immune homeostasis that is responsible. Okay. So what we have here in alveolar epithelium primarily consists of 3 types of cell. We have alveolar type 1 pneumocyte, alveolar macrophages, and alveolar type 2 pneumocytes. So there is an intricate crosstalk that happens between the macrophages and the type 2 pneumocytes, which is essential to maintain the pulmonary immune homeostasis. A part of the pulmonary immune homeostasis is to express chemokines to recruit monocytes coming from the blood. Now, these circulating monocytes play a very important role in the innate immune defense. So these monocytes, which have extra viscera, the fate of the monocytes depending on the environment and cues can either differentiate into an M2 macrophage, which is essentially required for the replenishment of the macrophages which is present in the lung. At the same time, they clear out the debris by autophagy. And number one is the M1 macrophage, or the classically activated macrophage, which is essentially required for killing of the cells. And the third is DC for antigen presentation. So what I'll be talking about today is Pseudomonas quinolone signal is a quinolone signaling molecule present in Pseudomonas aeruginosa. It is packaged in a bacterial membrane vesicle, and this quorum signaling molecule is actually the apex of all the quorum signaling, because mutating PQS leads to an avirulent strains of P. aeruginosa. Along with that, it also has the capacity to do cross-kingdom signaling. And two of the pioneering work based on which my research is based on that PQS is known to inhibit NF-kappa B and HIF1 alpha to translocate into the nucleus. So based on this premise, the question that we are asking is, can the bacterial membrane vesicle traffic PQS dysregulate the crosstalk between alveolar epithelial cells and circulating monocytes? So for this, we did a biofilm culture with 3 strains of Pseudomonas aeruginosa. One was a type strain and 2 clinical strains, and we did a 48-hour biofilm. So the first figure is a STEM picture of a biofilm coming out from a 48-hour culture. The second, we went on to analyze the bacterial membrane vesicles. We did a quantitative analysis with TEM, and we then went ahead with a quantitative analysis with NTA, which gave us the size diameter that all the strains more or less had similar sizes between 50 to 250 nanometer in size. Then we went on to detect PQS, which you see it here in acute of MS. So we detected this 260.1647 is the molecular mass of PQS, and we detected them in the bacterial membrane vesicles, which were isolated from the clinical strains. So what was interesting, that although all of these strains were coming out from 48-hour cultures, they produce different amounts of OMVs. So what you have here, that the type A strain, which is the PA1, has low amount of OMVs getting produced. The ATC, the other clinical strain, produced a mid-level OMV production, which was around 10 to the power 8 or 10 to the power 9 particles per ml, and the clinical strain produced higher amounts of outer membrane vesicles. Now, this correlated with the PQS concentration that we did by absolute quantitation on triple cord. So we found that those bacteria, which were the clinical strains which is producing more OMVs, were actually packaging more outer quinolone signaling molecules into these OMVs. So next, we wanted to simulate a lung epithelial model, and for this we use the gold standard, which is coming up with an air-liquid interface model with co-culturing type 2 pneumocytes, which is A549, and a macrophage, which is THP1. So what we saw that in an ALI system, and in the subsequent slide you'll see the reason behind it, is that when they're cultured together, so there are surfactant proteins which are expressed higher, which is not seen in the submerged 2D models. There is higher expression of cytokines and chemokines and CAPs. So we actually validated this model by looking at the markers, which is surfactant protein C and aquaporin 5, which was seen to be expressed in higher levels in this ALI model. Now, the reason behind this is the submerged 2D models that are coming up did not have the expression of chemokines, which was correlating to the VAP BAL samples that we are getting. And this was important for our subsequent study to look at the chemotaxis. Whereas the ALI co-culture was actually giving us chemokines which was at par with the clinical expression. So out of the chemokines that we were looking at, we were mostly interested in CCN2, which is MCP1. So what we saw that in LPS stimulated alveolar epithelium, there is increased expression of MCP1, both in mRNA and protein. When PQS extracts were added along with the antigenic stimulation, we saw a downregulation of these chemokines, which is CCN2 and CXCL8. We developed mutant strains of PQS, and we saw that this suppression was elevated and we had higher expression of the chemokines. Now, when this mutant extracts were supplanted by exogenous PQS, we saw that this this elevation was again suppressed. And we used an NF-kappa B inhibitor as a positive control. So we have an mRNA analysis on your left and CBA array analysis of the CCL2 and CXCL3 on your right. So what we are looking at, so the thing is these monocytes, these chemokines is important for the chemotaxis of the monocytes to be extravasaged into the tissue. So for that, we performed a chemotaxis assay. So we isolated PBMCs from the blood using FICOL, and then with a CD14 positive selection, we isolated the CD14 monocytes and went ahead with the with the transmigration assay.
We actually have 1 more minute.
Okay. So what we have seen that PQS inhibits chemotaxis of CD14 positive monocytes, primarily because there was lower expression of CCN2. So along with that, the next question that we were asking that what happens to the fate of these monocytes which was extravased into the tissue. So we started putting the PQS with M1 polarizing media, and we looked at 2 specific determinants. One was to look at the surface markers for M1 polarizing macrophages, along with that the cytokines. So we saw that CD80 and 86 were downregulated in presence of PQS, but the MHC2 was upregulated, and there was no difference in CD40. So we saw that PQS actually differentially regulated the surface markers. When it comes to the cytokines, we picked up three cytokines which were important for M1 polarization. We have IL-12A, TNF-alpha, and IL-10, and we saw that PQS downregulated IL-12A and TNF-alpha, but did not change the expression level of IL-10. So right now, the work that we are... just 1 minute. The time that we are doing is, so let me summarize. So the clinical strains of P. aeruginosa traffic higher levels of PQS. PQS suppressed the expression of major chemokines CCN2 and CXCL8 in LPS-stimulated alveolar epithelium. PQS-induced CCN2 suppression inhibits natural chemotaxis of CD14 plus monocytes. This leads to, and then we are right now currently working at PQS inhibition of monocyte into M1 macrophage differentiation. So this is essentially what we are talking about. As an acknowledgement, I'd like to thank my supervisor, Dr. Dipankar Ghosh, my senior who essentially started the PQS work, Ambalika Roy, who was a junior of mine, for helping me out, Dr. Jiva Shankar from AIMS for giving us all the Pseudomonas strain samples, Baba Saheb Ambedkar for the BAL samples, so Dr. Sumanta Basu for the CBA array analysis, for Dr. Priya Kanyan for the CBA array analysis, and DBT for my fellowship. Thank you.
Thank you, Priyakshi, for the clear presentation. We have time for a couple of quick questions. Yes.
Nice talk. Tell me, how do you know is your PQS pure? How do you know it's pure? Have you done studies to look at its purity, whether PQS is doing something or there's something associated with PQS?
Yes, sir. So when we have isolated the OMVs and extracted the PQS, we did an ethyl acetate extraction, and followed by we did an HPLC and then we just extracted the samples which was containing PQS, because you can actually, and then checked it out with MS, with LCMS, to see that it was only PQS that was coming in the extract and nothing else.
Nice talk. So do you know what is actually causing the hypervesiculation of your clinical strains? Because if you were to target that, you would were to block the QHS and also the other toxins and other things that are coming from the bacteria. So you have any inputs on that?
Right now we are doing a mutational studies to look at so what we have found out that we are screening around 9 to 10 strains of clinical strains of P. aeruginosa, and we saw that all the ones which are higher exporters of OMV is packaging PQS more. So we are doing mutation studies at different stages of OMV biogenesis to see what is the key reason because of which this OMV biogenesis is happening.
That would be interesting.
Just a small query. You said that the wild type and the typed Pseudomonas. Did you check the wild type, which one it is? Is it one among the typed ones also?
It is a PA1 type strain that we used it as a wild type Pseudomonas.
Okay, it's not there among the, the typed ones which you have chosen?
No.
Okay.
Okay, if there are no more questions, let's move on to the next speaker. The next speaker is Archana Padmanabhan Nair. She's from Narayana Nethralaya Foundation, Bengaluru. And she's going to talk about natural killer cells in macular degeneration.
What is the lab connector? We need that adapter. We have an adapter. Adapter कहाँ रख देते हैं ये? The mic is on. Just keep it off. Okay.
Good morning, everyone, and thank you, Immunicon, for this opportunity. So, in today's talk, we have two major players. One is the intraocular natural killer cells, and the second is the retinal pigment epithelial cells, and how their interaction is happening in the context of early age-related macular degeneration. So, until death do us apart. Age-related macular degeneration is a progressive degenerative disorder of the macula, and it is characterized by the presence of drusen and RPE abnormalities. And it is considered to be the second most prominent among different It is considered to be the second most prominent of the ocular conditions. And to the left are the different early symptoms associated. The most common one or the prominent one is a loss of central vision. And for this talk, I will focus more on the dry form of AMD. To the left are the different mechanisms that are associated with AMD pathogenesis, and our interest is in the immunoinflammatory mechanisms. The retina is a very complex system, and what happens in AMD is the accumulation of drusen, which activates the resident microglial cells in the retina, and that releases different factors as shown in the image, which calls different immune cells to the affected region. And this can be used for homeostasis, but when the condition goes worse, then all the homeostatic machinery breaks down. So what we would like to do or understand is, in order to understand the progression of the disease, monitor the patients with AMD based on the molecular biomarkers and associate them to the clinical phenotype, and also look at different methodologies where we can identify newer treatment modalities. As we all know, eye is an immune privileged site. Or is it actually an immune privileged site? So in order to for the eye to maintain protection of the ocular cells, it needs to maintain the immune balance. Now let me bring you to the concept of immune balance by bringing a corollary with the accelerator and the brake system. In case of an immune imbalance, there is a defective brake and there's a jammed accelerator. So with that concept, we hypothesize that there is a dysregulated intraocular milieu and it is disease causal in early AMD subjects. This slide gives you a study outline, and to delve further into the study design, this is the clinical arm where we collected aqueous humor during cataract surgery from patients and categorize them with those having retinal changes and drusen as early AMD and those without any changes as controls. The cells collected from the aqueous humor was used for phenotyping, and the cell-free aqueous humor was used for multiplex ELISA and untargeted metabolomics. So what are those immune activators that are present? We identified for the first time that there are increased NK cell populations in the early AMD aqueous humor, and that includes both cytotoxic and cytokine-producing NK cells. Furthermore, when we subdivided them as the activating to the activating NK receptors and the inhibitory receptors, we found the activating receptors to be increased in proportions. An unsupervised clustering based on FLOSUM showed us that there are 4 NK-related clusters, and the most prominent one was the cluster 1, which had expressions from an activated phenotype. We validated the finding in mouse model. We have a CRY-BA conditional knockout model where we did a single cell RNA sequencing and again identified the NK cell populations. That made us think that whether there is an interaction between the NK cells and the RPE cells. And just to summarize this slide, we did find very unique interactions, and 3 of them, like granzyme B, TNF, were all related to the cell death pathways. We further looked at the phenotyping in the mouse aqueous humor and again identified that along with monocytes and neutrophils, there was increased proportions of the NK cell subsets. This is a bit busy slide, but the take home from this is we did see NK effector molecules like interferon gamma, perforins, which were significantly high in the levels, and that showed a positive correlation with the NK cells from the same patients. Now, what are the dampeners present? We found that there is a reduction in the myeloid-derived suppressor cells in the early AMD aqueous humor. Furthermore, when we looked at the metabolite profile from the same subjects, there was a reduction in adenosine, considering that adenosine is coming, or the source of adenosine is from MDSCs. The same was observed in the mice model. So now the question is, does the dysregulated milieu affect the NK cell function? For this, we isolated the NK cells from healthy donors, treated them with the aqueous humor from cataract and AMD subjects, and in one set we supplemented with adenosine, then activated those cells and looked for interferon gamma levels. And what we saw was very interesting. There is a faulty immune dampening function when there is early AMD aqueous humor added. And on
supplementation with adenosine, we could actually regain that immune dampening function. The next question is, does an interaction happen between the NK cells and the RPE cells? And does it affect the RPE function, or it causes the death of the RPEs? For this, we cultured the RPE cells and polarized them in a transwell plate, added in a co-culture model, activated NK cells, and to boost the effect of NK function, interferon gamma, looked at transepithelial resistance and tight junction proteins by staining with ZO1. And we saw that on addition of NK cells, there was a reduction in the transepithelial resistance, meaning that the junctions are being lost, which was further confirmed by staining in these images. Moving on, we did see that with LDH assay and apoptosis assay, that there is a sort of death. So our curiosity peaked and we wanted to understand what could be the possible kind of death. And what you see is the video in a live cell imaging in a co-culture model of ARP19 and NK cell. And in the ARP19 cells, you can see vacuolations, increase in the size, and finally the shrinkage of cells resulting in death. And this is very prominent for a pyroptotic cell death. And the molecular mechanism that we looked at is by looking at the expression levels of Gas-dermin E. And we supplemented this system with adenosine. And as you can see, there is a reduction in the expression of these proteins. Also, the secreted levels, which is very unique to the pyroptosis mechanism, that is IL-1 alpha and IL-18, was also reduced. So the story so far, in an AMD human subject, we saw reduction in the proportion or increase in the proportions of NK cells, both cytokine producing and cytotoxic, increased interferon gamma and perforins, which are the effectors for the NK cells, reduced myeloid derived suppressor cells, and their effector, which is adenosine. And adenosine can suppress NK cell function. So with this scenario, we are seeing a connection with the RPE dysfunction and death by pyroptosis, and supplementation with adenosine can inhibit this feature. So there are many questions that still needs to be answered, but what we think would be a therapeutic relevance is where we can look for newer treatment options, where we can look at NK cell modulation and also look at adenosine supplementation. So I would like to thank our funding agencies, which is Naren Netralia Foundation and Arvo Foundation for Eye Research, my mentors, without which this study is not possible. And we call ourselves growers. These are my fellow workers. And the clinical team at NN, Professor Debasishan Sinha, he was the person who helped us with the mice model. And last but not the least, the patient volunteers. Thank you.
Thank you, Archana, for the nice presentation.
Yes.
Any other immune cells which is
There in the aqueous humor in AMD?
Yes, ma'am. So we looked at, so based on existing literature, we know that there are monocytes that infiltrate. There are the resonant glial cells, which can again convert the monocytes. But in the aqueous humor, we found neutrophils. Then we also found B cells, T cells, but more prominently, we were seeing the innate cells, which were...
Well, because the complement regulatory defects are very much associated with the AMD.
Yes.
So I was thinking whether NK cell mediated ADCC is also having a role in the injurious processes.
That could be a possibility, ma'am, because what we do know, I mean, I don't have data to suggest, but literature does say the presence of all the HLA related molecules on the surface of RPEs.
Okay, thank you. I know you are not working on those lines.
Sure.
Have you noticed any activated CD8 T cells there?
So in the intraocular milieu, what we are seeing is that the T cells are less abundant compared to the innate cells. This is a speculation I have, that there is an early calling of the innate cells first, and probably maybe over the time of the disease or in the progression states, maybe you would actually find T cells in the active state.
Nice presentation, Archana. I want to,
Yes, sir.
I want to ask you if you see this earliest changes in the retinal pigment epithelium in human specimen
because you have an option to get a human, just a rip of the retinal, after retina, you rip off you see the changes there.
Yes, sir.
I would be interested before NK cells involvement, the autophagic mechanism that is being playing the role. Okay,
Yes.
that would be both in the mouse model as well as the human model, give much more evidence for the early changes.
Yes, so you're absolutely right, sir. So there are what happens in AMD in the disease pathogenesis is that when I said drusen formation is because of the lack of clearance, which means there is a dysfunctional autophagy already involved in the process. So that is why the clearance of these drusen components does not happen. Now, even in the mice model that we have, which is when I say cry-BA knockout model, it is basically a model where the crystalline protein is not cleared out of the system, and that causes the RPE pathology. And that's said for your second question, do we see changes in the retina in early AMD? So this is basically the image of an early AMD subject. The irregularities you see, these are the different layers of the retina, and you see irregularities, especially in this bright layer, which is the RPE. So we do see irregularities.
Yeah, for crystalline again, alpha, beta, gamma,
Yes.
the gamma crystalline will be much more important for those things.
Will
Thank you.
take into consideration. Thank you, sir.
In the interest of time, can we continue in the tea break, please? Already we are delayed by 25 minutes.
Thank you.
I request Professor Lotpal to continue the session.
Our next student speaker is Vandana Krishna. She's from BITS Pilani, Hyderabad campus. And she'll be speaking on development of surviving mRNA-loaded lipid nanoparticles for oral cancer immunotherapy. Vandana. So I'll buzz you after 7 minutes.
It's not working. It's not working.
It's not connected. This way.
Can you try?
Good morning, everyone present here. I am Vandana Krishna from BITS Pilani Hyderabad campus, a PhD student pursuing under the guidance of Professor Venkatavamsi Krishna Venuganti. The topic for today's presentation would be the development of survivin mRNA-loaded Development of survivin mRNA-loaded lipid nanoparticles for oral cancer immunotherapy. Survivin is the smallest protein which belongs to the family of inhibitor of apoptosis, and it has a role in cell division in a normal cell. And it is a tumor-associated antigen since it is overexpressed in tumor cells. And immunohistochemical analysis of this particular protein in oral squamous cell carcinoma and pre-neoplastic lesions show a significant overexpression of this particular protein. So when this protein is present within the cell, it undergoes cleavage, and one of the peptide form of this particular protein gets expressed onto the cell surface of the tumor cells, which is recognized by the T cells, and it activates the T cells, causing the T cell to get activated, and in turn causes the cytotoxic. In turn causes the lysis of the tumor cells. So the objectives of this study was to first check for the survivin expression in oral cancer cells, and then to figure out the immunogenic fragment of this particular protein and clone it into a plasmid, and then produce an mRNA using the in vitro transcription method. Further, considering the instability of a naked mRNA, we wanted to load them into nanoparticles using a microfluidic device. We have used ionizable lipid of ALC0315, and further characterization of this nanoparticles were done. And to evaluate the immunogenic and anti-tumor activity, we first did the cell uptake mechanism in bone marrow derived dendritic cells, the translation of mRNA to protein, and the proliferation and activation of T cell population, cytokine analysis, and the anti-tumor activity. So in the upper panel, we see that this particular survivin protein is overexpressed in both FADU, which is a human-derived oral cancer cell, as well as in MOC1. MOC1 was used for all our animal studies, and hence the expression was checked in that particular cell as well. And we see that it is present both in the nucleus as well as in the cytoplasm of the cells. And in the below panel, we see that it is the design of our mRNA, which has a promoter of sp6 and a 5' untranslated region, and the immunogenic fragment of survivin, which is of about 45 nucleotides, a linker, and a histidine tag, as well as a stop codon, followed by the 3' untranslated region and a poly A tail of T2 nucleotides. Post-cloning, we synthesized the in vitro transcribed survivin mRNA, and we checked for its integrity on the denaturing acrylamide gel as shown. The complete size of this particular mRNA is about 415 nucleotides. And to synthesize the mRNA-loaded lipid nanoparticles, we used a droplet-based microfluidic device where the organic phase consisted of ethanol, lipids, and the aqueous phase consisted of our mRNA and tween in the concentration of 0.05% to reduce the particle size. We see that the organic phase and the aqueous phase are both perpendicular in their streams, and at the interjunction of the points, they form a self-assembly of liposomes, helping in the encapsulation of survivin mRNA-loaded lipid nanoparticles. And the particle size was about 300 nanometer post encapsulation, where with the polydispersity index of about 0.12. And the below panel, in the first image, we see the stem images of the mRNA-loaded lipid nanoparticles. And in the second image, we conducted a decomplexation study to confirm that the mRNA had actually complexed with the lipids of our interest, and it is not present on the outer phase of the formulation, for which in one sample, we directly loaded the mRNA-loaded lipid nanoparticles, and in the next sample, we used a decomplexing agent such as dextran sodium sulfate to decomplex the complete structure, and then we ran it on the gel to confirm. We see a band of mRNA on the next lane of the gel. And to further confirm that the mRNA-loaded lipid nanoparticles actually provide stability to the survivin mRNA, we did something called as stability assay, where we subjected a naked mRNA with RNAs, and we see complete degradation as seen in the third image C lane. And we subjected the same like RNAs with mRNA loaded lipid nanoparticles, and we see very faint smears suggesting that the mRNA loaded lipid nanoparticles actually do provide stability. And then we carried out with cell uptake and translation of mRNA. So for the cell uptake study, we tagged our mRNA with cyanide-5, and we checked the cell uptake in bone marrow-derived dendritic cells, considering the concentration dependent, and we also checked for the naked as well as mRNA loaded lipid nanoparticles in 2 different concentrations. And we confirmed that the uptake is actually concentration dependent of the mRNA, and the mechanism of cell uptake was mainly through clathrin, actually a combination of caveolin, clathrin and macropinocytosis, but majorly through clathrin and macropinocytosis pathway. And since the mRNA was synthesized in the laboratory, we wanted to confirm that it is actually translating to a protein, for which we picked human forearm fibroblasts, because these have very basal levels of survivin present for their cell division function. And we have a control group and a treatment group where we see an upregulation in both survivin and polyhistidine levels.
You have 1 more minute.
Yes, survivin and polyhistidine levels, confirming that it is actually translating to a protein. And when we checked for the in vitro proliferation of these activated cells, we see higher upregulation of the CD8 plus T cells and the activated T cells represented by CD8 plus and CD25 plus. We also noticed an upregulation of T regulatory cells, but this particular population was much lesser compared to the CD8 plus or the activated T cells. And the in vitro cytokine analysis also showed that there is significant higher levels in the groups of CTLA-4 inhibitor, the group treated with survivin mRNA lipid nanoparticles, and the combination of the two. And further, we checked for the cell viability of cancer cells and healthy cells to check for the anti-tumor activity. And it is seen that the group of survivin mRNA and the combination of that with CTLA-4 inhibitor showed drastic reduction in the cell viability. And the healthy cells which were present in the culture did not have any reduction in the in their cell viability, even though the protein is expressed in them. This could be because this protein is particularly present only within the nucleus of the cell, and compared to the cancer cell where it is present on the surface. And when these nanoparticles were administered into healthy mice, we saw enlargement in the spleen and the lymph nodes. And to conclude, this study successfully synthesized mRNA-loaded lipid nanoparticles, and the expression of tumor-associated antigen in the cancer cells could be critical for effective immunotherapy. And the in vitro assays revealed that significant anti-tumor activity. I would like to acknowledge my supervisor, Professor Vamsi Krishna Venuganti, in my campus, BITS Pilani Hyderabad campus, the funding agency DST SERP, and the organizers of IMMUNOCON for giving me this opportunity. Thank you.
Thank you. Maybe a couple of questions.
So in your in vivo study, mice check whether they are really targeted to antigen-presenting cells?
Under in vitro conditions, we have checked that it is actually upregulating MHC1 and MHC2 complex compared to the macrophages. And because of the particle size, it is preferably taken up by the dendritic cells compared to the macrophages, because the particle size is around 300 nanometers. Under in vivo conditions, we are yet to confirm the same.
Any more questions? Yep.
Have you worried about the immunogenicity of the lipid? Because when you say this to a student, he says lipid. I said yes. In tuberculosis patient, we find tons of antibody against lipids.
Yes.
So lipids are immunogenic. So when you use lipid and mRNA nanoparticles, have you worried about the immunogenicity of lipids?
We have checked for its effect, specifically with the T cell population, and we have also checked its effect when administered into the animal. Like specifically in this particular slide, we do see that blank lipid is also enlarging the localized lymph nodes and the spleen also to a certain extent, but they do not show any significant upregulation in any of the interleukins, or they do not show any anti-tumor activity related. But further safety studies in the animal model is to be done.
Okay, maybe we'll move on to the fifth talk of this session.
Thank you.
Thanks, Vandana. Next talk is by Akanksha Chauhan, and she's from Aisar Mohali, Punjab. And she'll be talking on investigating the multifaceted role of Vibrio parahemolyticus, got it? TDH in modulating macrophage function, a study of inflammation, polarization, and cellular signaling. Akanksha.
So very refreshing morning to each and all present here. My name is Akanksha Chauhan, and I'm going to present a part of my work, which is to investigate the multifaceted role of Vibrio parahemolyticus thermostable direct hemolysin in modulating macrophage function. So, talking about the pathogen of interest, which is Vibrio parahemolyticus, it's a human gut pathogen and is a major foodborne pathogen which causes severe acute gastroenteritis, and in some immunocompromised individuals, septicemia can also be seen. Among all the virulence factors that it possesses, the thermostable direct hemolysin, that is TDH, has been shown to be a key player in the pathogenesis. By nature, it's a pore-forming toxin, and as the name suggests, as soon as it encounters the membrane, it punches hole onto it and do the cytolysis. Now, talking about the relevance of the study, so over 3 billion people globally depend on its seafood as their major staple of diet. And talking about the past outbreaks in this field, so there have been cases from 1990s and latest to 2020, and the number of deaths reported are increasing with time. So Asia also reports the maximum number of cases annually. So that's why the study of this pathogen becomes quite important, and we are interested in knowing how exactly modulating the host immune function. For that, we did our experiments in vitro and ex vivo in raw and BMDMs, and we treated the macrophages with the particular protein and looked for the inflammatory response. Apart from that, we have also employed the in vivo studies in knockout mice. So to start with, we checked the cytotoxic response. So we treated the macrophages with protein and looked for the cytotoxicity, and with a very low concentration also, the protein was able to induce cytotoxicity. Apart from that, we have also checked for the pro-inflammatory markers, mainly cytokines. Then we have also looked at the reactive oxygen species, as well as nitric oxide production, BMDMs. And all this suggests that it indeed induced potent cytotoxic response as well as pro-inflammatory response in macrophages. Now we were interested to look at which particular macrophage subtype it exhibits. So in one of the markers for M1 and M2, which is iNOS and arginase. iNOS was highly upregulated at 12 hours of time point, which downregulated at 24 hours, while opposite was seen in case of arginase, which suggests that probably at initial time point, TDH is exhibiting M1 type of polarization, which is later on is doing a macrophage polarization switch towards M2. So apart from that, the next question was, what are the receptors involved? So we first started with exploring TLRs, which are the major PRRs, which are widely studied in the bacterial ligands. So we employed ex vivo studies, and we looked at IL-6 and IL-1 beta release with the receptor knockout conditions. So we used TLR4 as well as TLR2 knockout mice and looked for the cytokine release, and we got a abrogation of the cytokine levels in both of these knockouts, while TLR1 knockout showed no decrease. That means indeed TLR2 and 4 are there for the TDH recognition. Apart from that, for checking the physical interaction, we employed co-immunoprecipitation studies, wherein we got the pull down of both TLR2 and TLR4 with TDH, which tells us that TDH physically also interacts with these two TLRs and induces the pro-inflammatory signaling. Apart from that, the next question was to deduce the underlying signaling pathway. So we employed inhibitor studies and we probed for the key molecules which could be involved in the process, like transcription factors NF-kappa B-AP1, MAP kinases P38, JNK, and ERK, as well as other signaling molecules like PI3K and AKT. So I'm not showing the data. So to summarize with what we got is the differential involvement of all these molecules in induction of different cytokines. So as we know, signaling is such a complex interplay of molecules. It's never a direct process. So the interesting or unusual part which we can take from here is that ERK has been shown to be negatively regulating TNF-alpha while positively regulating IL-6. Also, PI3K-AKT, which is a conventional, which works in a conventional same pathway, has been shown to work independently in regulating TNF-alpha and IL-1 beta. So these two things that we are considering, and we are narrowing it down to pursue further. And we wanted to know how these molecules are differentially inducing the cytokines, but how exactly these downstream molecules upon activation can regulate polarization, is ultimately leading to inflammation, is still a question, and that still needs to be explored. Further, we extended our studies towards in vivo model. So what we gave intraperitoneal dose of TDH in mice, and we checked for the pro-inflammatory serum cytokine levels, basically, in terms of IL-6, IL-1 beta, and TNF-alpha release, and we got them. Apart from that, we were interested to know, are the receptors which were involved in macrophages, are the receptors same at systemic level too? So again, we employed the knockout studies and looked for the IL-6 and IL-1 beta release, but this time in serum cytokine levels. And again, we got the same TLR2 and TLR4 involvement at the systemic level too. So the one interesting observation while we were doing these in vivo studies that we got was the serum when we were collecting that was hemolyzed, that has the RBC's contamination, and a condition called hematuria, which is we can see a blood in urine. So then we wondered why this protein is causing so much damage to the mice. So then we were intrigued to look at the health of the internal organs. So when we dissected the mice, and as the target organ, intestine was quite expected that it could be inflamed, but we were very surprised to see the damage which was linked with the other vital organs too, like spleen, kidney, liver, and heart, which were enlarged in size, they were damaged, they were highly inflamed. So then, when we digged into the literature, we could find one particular syndrome, which is hemolytic uremic syndrome, which is till now has been shown with E. coli, Shiga toxin producing E. coli bacteria, but has not been shown for any of the Vibrio species. And so further, we would like to characterize by using this triad of symptoms, which is hemolytic anemia, which it possesses. Thrombocytopenia is the loss of platelets that we are still seeing, and acute kidney failure. So we are doing this kidney renal function test. We are measuring urea creatinine level, and we are in process of characterizing it. If it is Hus or Hus-like symptoms, is it showing?
We have 1 more minute.
Yeah, sure. So far, it's at ex vivo level in macrophages. What we have shown. Till now, there's not been any receptor which is known for this TDH, but we got the TLR4 and 2, which is involved in its recognition and is inducing pro-inflammatory response generation cytotoxicity in terms of all the pro-inflammatory mediators release. We are also getting the macrophage polarization in terms of IL-10 release, as well as INOS and arginase being regulated differentially at different time points. Apart from that, what we got at in vivo level, that there were vital organ damage when it was given at in vivo level, and there were Hus or Hus-like symptoms that we still need to characterize, and we are very interested to know the mechanism behind it, because the report suggests that complement dysregulation, genetic modulation, and there are probably many other factors which could be involved in initiating this. But how exactly it is happening, we don't know. With respect to the genetic modulation and the factors taking part in it, we have some other observation which is not making sense, but we hope that in future will make some sense. So TDH itself has been shown to translocate to nucleus. And also, it is also linked with P53 activation that we have seen. So So all these thing is making any sense, or is it is connected to each other, that we are still in process of figuring out. So we are joining the missing links. So it's under construction. Yeah. So in the last, I would like to acknowledge my supervisors, Professor Arunika Mukhopadhyay and Professor Kaushik Chattopadhyay, my lab members, and ISO Mohali and all the funding agencies. Thank you so much.
Thank you. Thanks a lot. The talk was as excellent as the timing. Questions?
So here you have one ligand, but seems to be interacting with two receptors.
Yeah.
But if you take two very defined ligands, let's say for TLR2, TLR4, do you think you are going to get the same finding?
In case of TLR4, we have checked with competitively, so with LPS, so it still shows the response. But with TLR2, we are quite in mid of finding answers to it, because if you see the data also, TLR4 shows complete abrogation, but TLR2 is not showing the complete abrogation. It is showing a decrease, but it's a partial decrease. So it's having maybe some receptors interplay or some partial role in induction of the process that we are looking into it. Yes, ma'am.
So good presentation and good discussion. I think you can try to explore the complement activation stages and complement regulation.
We are planning, ma'am.
I think you will get a quick answer also.
True. Yes, ma'am. Yes, ma'am. Thank you so much, ma'am.
Any questions from the students? This is by the students of the student session. So, okay, you can go ahead.
Hi. Very nice work. So I'm here. So this molecule is interacting with both TLR2 and 4. That's what you're saying. So it is giving you a mixed result, because TLR4 gives you a different kind of pro-inflammatory pathway and cytokines, and TLR2 gives you perhaps more like anti-inflammatory also. So I think doing an experiment in TLR4 knockout versus TLR2 knockout independently, then you can get a clear picture. Because right now what is happening, you are putting this molecule and you're getting everything mixed. And the second thing is, if you have a knockout or something, or you can block the MYD88 and see what is happening. And
We have checked with the migrated knockout mice, and we get no responses there. Yeah.
And maybe if you go more mechanistic, using some sort of structural approach or computational approach,
Yes, sir.
and to see what you can modulate in this, so it can become binder of this, but not this.
Right.
So what are the common things? So I think these
Yes,
are
sir. Yes.
very nice.
Thank you so much for the suggestion, sir.
Okay, perhaps in the interest of time, we will go to the last talk of the session. The speaker is Suraj Singh from IIT Palakkad in Kerala. Suraj?
Thanks to the speakers and the chairs. I would request the chairs of the session, the speakers of the GP Talwar talks, and the speakers of the oral presentation talks to kindly come up on the stage for a group picture. I request the speakers of the GP Talwar talks also to kindly come up on the stage. I request the GP Talwar talk students also to come up on the stage if they are present in the hall right now.
All of them are girls. I can see all of them are guys. Good. I don't think a photograph will take it.
Dr. Nindi, please come, please.
No, we'll have more opportunity.
Congratulations.
Thank you, everyone. Now we will have a short break for tea, and I request everybody to kindly reconvene by 11:50. I also
an outsider's reflection on immunology. I request Professor Santosh Kar to chair this session and introduce the speaker.
It is afternoon, I think just becoming afternoon, 12 o'clock. And it is you can't imagine the pleasure that I have to meet Balram, whom I know for last 52 years. 52 years, more than that, I think. Because he was a student in IIT Kanpur, chemistry department, doing masters in chemistry. I was doing my PhD there, and we were together in the same lab. T. Balasubramaniam and Professor S. Ranganathan, Professor Balasubramaniam and Professor Ranganathan were sort of nurturing him, tutoring him, mentoring him. I was not being, nobody was able to mentor me. I was a little aggressive. As you can see from now, if at the age of 80, I'm so aggressive, you can imagine at the age of 23 how much aggressive I would have been. But we were great friends because Balram used to be very, very quiet at that time. I have taken permission from him to talk about a little bit about him. When he got married to Hema, I was there. When his son was Aditya was born, I was there. I visit his house when he was not director of Indian Institute of Science, and when he was director of Indian Institute of Science, he will make a dosa for me and eat. We will eat. But the good thing is, we never talked about anybody else. We talked science. We talked about research that was going on. Whenever I visited Indian Institute of Science, if I have time, he will ask me to give a talk to his students about immunology. I remember the present director, DBT secretary, Rajesh Gokhale, yes, wanted to make an antibody against a peptide. For his PhD work, he was a student of Balram. Balram said, Santosh, look, he's asking about how to make antibody against peptide. I know how to make peptide, but can you tell him how to make antibody against that peptide? So I sat with Rajesh and explained to him how to conjugate the peptide to a carrier and immunize and how to purify antibody. Now Rajesh Gokhale went from here to Stanford, came back to India, joined NII, and then now he's the DBT secretary. You can imagine standing here in Indian Institute of Science, trying to introduce Balram is not necessary. Everybody knows him about him. Only thing is that I can say some chutkules. He was a student of MSc. I was as active as anybody in IIT Kanpur. We had invited G.N. Ramchandran to give a lecture. And Balram is standing behind, said, Santosh, can you introduce me to G.N. Ramchandran? I was the person who introduced him to G.N. Ramchandran. But then when he came back to Indian Institute of Science, you know what happened. I don't have to tell you. Ramchandran and Balram were probably part of the same coin. Every day I know GNR will remember Balram, and if possible, he will go there and meet him in his office or at his home. Balram, actually I mentioned that just now, but I will not venture to do that. We should have given him the best teachers award in immunology. Whether it is immunology, molecular biology, protein chemistry, anything, there cannot be a better teacher than him. His lectures are legends. I just now took his permission that if I come to Bengaluru, because I don't want him to travel, my student is the director of this Mahipal Institute of Integrated Biology or something, Jyoti Prasanna. She said, sir, please come to my institute and do with students whatever you want to do. I have got the acceptance by Balram that he will travel from his home to this institute and interact with students. My point is that this type of people, we need to create more. We need to give hopes in the younger generation that science is only going to solve the problem that we are facing in our life. Be it filariasis, because I worked on filariasis, or be it worm infestation of our younger generation. Believe me, 25% of 200 million children who go to school are worm infested. They are carrying ascaris in their tummy, and we give them albendazole, a drug which was made for filariasis, a drug which is highly toxic. We have never bothered about doing research on that. We must do research. And I'm sorry, I'm coming into my domain now. My point is that let us listen to him. Well, it is like rasagulla. For me, it is like my best favorite food. I have listened to him 200 times maybe. And when he writes a article recently, a few years ago, he wrote an article about IIT Kanpur, and he sent me a request that do I have a photograph of Professor Ranganathan, S. Ranganathan in the olden days. I said, no, I do not have a photograph. But he managed to get something from his son, I think. Professor Ranganathan is dead, but Anand Ranganathan, and I think he managed to get it from him and publish that article. You all should read that article in Current Science about Indian Institute of Technology, IIT Kanpur teachers. So my point is that it is like, no, I'm sorry, I will take 1 more minute. It was like in, Balram will justify, Indian Institute of Science was celebrating 100 years, several years back. They invited Sidney Brenner, my guru from Cambridge, as the chief guest. Sidney Brenner came. He doesn't travel, but he came. I'm sorry, I'm half a more minute. Obaid Siddiqui was alive and introduced Sidney Brenner and talked for 45 minutes. 45 minutes it took him to introduce Sidney Brenner. And Sidney is sitting. I'm also in the audience. I flew from Delhi to Bengaluru. And then when Sidney's time came for speaking, he said, Ubaid has said everything about me. Any questions? So my point is, I'm not going to get into that situation. He's a legend. He should be respected and listened to. Thank you, Balram. Please come.
Okay, good afternoon. Is this working? Yeah. I'll try to be as brief as I can so that you make up some lost time. I'm really not an immunologist. I'm here because Professor Nandi, I can't say no to him. He said that there will be only a few students left on a Sunday afternoon, and so why don't you come and talk to them? So here I am. What is my connection with immunology? This slide tells you a little bit about that, because when I went to the United States in 1969, and was looking for a problem to work on for my PhD, my supervisor told me that maybe you could use the newly developing techniques of NMR spectroscopy, which were just beginning to be applied to biology, and maybe you could use them to study hapten-antibody interactions. Now, I did not know what a hapten was, and I did not know what an antibody was, but I began to read. And the first book that I looked at was this book, which was available in the library by Karl Landsteiner, on the specificity of serological reactions. It was fascinating. Not only was it fascinating, but the entire idea of studying hapten-antibody interactions at a molecular level was really something that interested Linus Pauling at that time. And Pauling, of course, was legendary. He was a hero to every student of chemistry. And therefore, Pauling was really divine. And so anything that he said must be worth following. It then turned out, I realized that if I were to make antibodies, I would have to inject a protein, albumin in those days, conjugate it to a small molecule, and inject it into a rabbit. And so I did go along to this lab where they were doing these kinds of experiments, but then I realized that I wouldn't be able to hold a rabbit and inject a rabbit at all. And I wasn't made for this. So I slowly migrated away to other biological applications of NMR spectroscopy at that time. But over the years, as I became interested in protein structures and biochemistry in general, I found this one day while teaching. This is on the Pauling blog, and this is a copy of Landsteiner's book, and this is annotated by Pauling approximately in 1936. And in 1936, Pauling writes, I think the protein must contain side chains with reactive groups. I thought that was marvelous. 1951 was when Pauling would give the alpha, tell us all about the alpha helix, and it would take another few years later that Max Perutz and John Kendra would show the first structures of proteins. Pauling, of course, was interested in immunology. Pauling made two very famous errors in his career. He was right so often that he could afford to be wrong in these. The first was in his theory of the structure and process and formation of antibodies, which he wrote in 1940. A long paper in the Journal of the American Chemical Society, and this advanced what was then called the instructional theory of antibody formation. This was many, many years before McFarlane, Burnett, and others got the kinds of theories that you talk about in immunology today. His other famous mistake, of course, was the structure of the nucleic acids, of DNA in particular, published in 1952. This was a marvelous error, and students should know this, because this was Pauling's triple helix, but it had all the interactions, all the salts, all the charged groups inside the structure. So the structure should have just exploded. And the leading expert on ionic interactions in the world in those days was Pauling himself, but he made that mistake. So my subsequent brushes with immunology are really due to these three people. Professor Talwar, who I brought the slide along specifically because he's here right in the audience, and I show you two of his papers, one in 1976 and the other in 1982. And in the 1970s, I had just joined at the end of 1973, the Indian Institute of Science. I was just setting up a laboratory. In those days, we did not have too much by way of facilities. And in 1976, I got a letter from Professor Talwar. Apparently, one of my mentors at Kanpur had suggested that he write to me. He wrote to me, asking me something about whether a student of his could come and work on a peptide that they were making, and he brought the peptide and came along. This was the student S. Ramakrishnan, who's an author on this paper in PNAS in 1976. So he brought along a poster, and this poster had the structure of HCG, and I show it to you on the next slide. But Professor Talwar's work really focused on making peptide antibodies. And this was to develop a contraceptive vaccine in which the antibodies would capture HCG, and therefore the pregnancy would not proceed further. The remarkable thing about this work was that he had recruited at that time Dr. K.S.N. Iyer, a peptide chemist, to his group. And Iyer began to do solid phase synthesis, which was not really accessible in those days. For those of you who are technically inclined, solid phase synthesis, after Merrifield first published it, was rather difficult to do, both because of the nature of the polymer supports and the kind of protecting groups which were there. You could make the peptide, but you couldn't get it off the polymer very easily. And the last step in that reaction required anhydrous hydrogen fluoride. And it turned out that in those days, everybody would make the peptides on the polymer and then send it across to Japan, where Sakakibara would then use anhydrous hydrogen fluoride and then send back the deprotected material. So there you are. This was the picture that I saw on the poster, which Professor Talwar brought. It didn't have the disulfide bridges in HCG the right way. The glycoprotein hormones turned out to be far more complicated than we imagined what they were in the 1970s. And what I've boxed in red is what I saw. Professor Talwar's student has connected those two cysteines there in a disulfide bridge. And I was a peptide chemist. I was in G.N. Ramachandran's group at that time, and so I was looking for problems to do. So I thought we could make this very small peptide, disulfide linked. It might be antigenic, it might be structurally very interesting. Such small disulfides had not been studied at that time. And so a student of mine embarked on this project. Took us a while to work on this. We worked out the structure and everything. And then it turned out that this paper appeared from Professor Bell's lab in 1982, which gave all the disulfide bridge pairings. And it turned out that that disulfide bridge was not there at all. So here I was with a peptide, which I'd studied in great detail. Now, if I wanted to write a paper, I must, of course, impress the referees with the biological importance of the problem that I'm addressing. But now the biology has disappeared because the disulfide does not exist in HCG. But as luck would have it, Arne Holmgren in Stockholm at that time had just worked out the sequence of the thioredoxins. And this whole class of redox-active disulfides, all of them had the same kind of two residues intervening between the two link cysteine. So all that I had to do then was to change the introduction and say that I am now making models for thioredoxin. And this, of course, one got a large number of papers for me in subsequent years and even got me a trip to go to Sweden to attend a conference. But look at that at the bottom. When the crystal structure of HCG finally came out, it turned out that was in 1994. So you can see the difference, 76, 82, 94, the decades are passing, and the cysteine knot was discovered. The cysteine knot had just been discovered a little bit earlier in the growth factors. Epidermal growth factor was the first one. I later on did have a brush with immunology with Professor Radiga, who used to work here on antibodies to neutralize riboflavin carrier protein and once again terminate pregnancy in rats. And one was looking at the possibility of enhancing structure. But today I haven't really come to talk about my work. I've really come because that has no connection really with immunology. But I've really come to tell you what I think as an outsider about immunology. I want to show, if the students haven't seen this, to introduce you to two papers. One which I saw almost when it appeared and I was fascinated by it. This was in Nature in 1987. This was Don Wiley's paper on the MHC with unresolved electron density at the site, at the binding site. There was a peptide bound already on the MHC for presentation, but one did not know what that peptide was when the structure was solved. The second paper on the right is again from Don Wiley, Nature in 1981, which is the hemagglutinin structure, the influenza virus hemagglutinin, a glycoprotein on the surface, very important for viral infection, and there's the structure. I'm going to show you a little bit about this. But I also want to tell you a little bit about why these papers are important for immunology. They're important because you can see what Steve Harrison wrote after Don Wiley died prematurely in 2001. he wrote that how they imagined that they would now develop structural molecular biology as opposed to the informational molecular biology that was then reigning in Cambridge, Massachusetts. So Harvard was full of the information molecular biologists. All institutions still are. But today, of course, many times they've been overwhelmed by the power of structural biology. But the MHC structure was remarkable because the MHC structure showed eventually, as it was resolved to higher resolution by 1994, showed you how the peptide was bound to MHC. And for the first time, we saw a peptide bound to a protein in which the side chains had very little to do with it, and very few side chains. Everything in the middle was pointing outwards, waiting then to bind to the T-cell receptor. And some years later, Wiley published the structure of the complex. Today, when you look at this kind of antigen processing which you have, you can get these cartoons like this. It is these cartoons which push anyone who doesn't like complexity out of immunology. And now, but if you look very carefully at it, what is happening? It's really carefully regulated proteolytic processing. Proteolytic processing lies at the heart of antigen presentation. And this kind of processing requires the evolution of proteases, and one might ask questions, how did the immune system really evolve to this complexity? These are the kinds of questions that I think one ought to ask in future. But I'm going to show you what Wiley did again. The influenza hemagglutinin, he got the structure in 1994 at a pH at which it fuses with the host membrane. And then when he got that, there was this remarkable structural transformation where the fusion peptide simply came out. So you can see what's in yellow here is in yellow here, what's in red here is up there. These are drawn so that they are more or less aligned with one another. This is the remarkable transition of the surface viral protein, which results in host virus membrane fusion. This opened up the field of virology. So in fact, two subjects, immunology and virology, now really became connected to one another. And today you will see, of course, that every time you talk about a virus, you're also trying to neutralize that virus with a vaccine. I have only two more slides. And here you are. I call this viral host fusion in Wonderland, because this is what happens just like Alice drinks something and goes from big to small. That's what happens, for example, to the coronavirus spike protein. Everybody's forgotten the pandemic now, and that is why I want to remind you about it. And everybody's heard of the spike protein. This is the pre-fusion structure of the spike protein. This is the post-fusion structure of the spike protein. And for the first time last year, Bing Chen at Harvard has published the structure of the post-fusion structure of the coronavirus spike protein. This is the first membrane protein where the transmembrane domain, the fusion peptide, all of them are available. And this is now going to give us a new view of virus membrane fusion. This will be applicable now to all the enveloped viruses, certainly applicable to influenza and HIV. But this enormous conformational transition which takes place is possible because of proteolytic removal of this cap. And so you have a loaded spring, which is the metaphor which the virologists use, which then extends outward to induced fusion. Is this important? It is, because what does immunology promise everybody? Immunology promises everybody a life free of disease at some point or the other, because you'll have a vaccine for every disease. And of course, the world has spent an enormous amount of money on the COVID-19 vaccines. The question, of course, which one might ask is, how effective were the COVID-19 vaccines in controlling the pandemic? Or did the virus really adjust to the human host in a natural process which Darwin might have envisaged a century and a half ago? This is a paper which is the architecture of the inactivated SARS-CoV-2 virus. And that's what you do if you want a viral vaccine. You inactivate the virus. What do you inactivate the virus with? You inactivate it with this organic compound, butyrolactone. What does butyrolactone do? Butyrolactone reacts with amino groups, primarily. And it certainly reacts with all the primary amino groups on the lysine residues of the spike protein on the surface. And it therefore turns out that the spike protein undergoes the conformational transition once this happens. And what you have are post-fusion spikes, which you can see by electron microscopy. But you don't want antibodies to a post-fusion conformation. You want antibodies to a pre-fusion conformation so that you can capture the virus before it has fused with your membrane. And in this paper, which appeared, for example, in Structure, they say the study calls for crucial structural characterization of vaccine candidates. Now, of course, the Russians have published a paper on the Sputnik vaccine, which uses the same procedure for inactivation. And now it turns out that they conclude in this that electron microscopy is valuable for quality control of the vaccine. They also say that they conclude that the butyrolactone procedure is not good enough to be proposed for inactivation of the COVID-19 vaccine because of some peculiarities of the spike antigen. Those peculiarities are they're not peculiarities, they're standard protein chemistry. And why am I telling you this? Because if you read the newspapers, you will find that there is now a raging controversy about a paper which appeared from the Banaras Hindu University on the apparent toxicity of the COVID-19 Covaxin, which is a butyrolactone neutralized vaccine. But of course, there is a legal case which has now been filed by the company against those who wrote the paper. The paper is a bad paper. It doesn't have the proper controls. But one should have otherwise asked the question, is there any quality control for the vaccines that are produced like this? And these are, I think, the kind of issues that immunologists might actually look at in future when they promise vaccines for all the new and emerging diseases which we will undoubtedly encounter in the future. I think that's stopped, and I will stop at this point because the last slide has nothing except to say that I am no, I think it's gone to sleep, doesn't matter. I just want to conclude with this slide because one needs to understand chemistry, which I studied, as Santosh said, and one needs to understand chemistry well whenever one deals with proteins. And Arthur Kornberg, the man who discovered DNA polymerase, fired the first shot of the biotechnology revolution, said that chemistry, in his old age, he said that chemistry is the lingua franca of the medical and biological sciences. And I thought it might be useful in my brief presentation to remind all the students of this advice which Kornberg really has given. Thank you. If there's time, I don't want to delay anybody. Yeah.
So you said Linus Pauling got two things wrong. What do you think of his assertion that vitamin C is good for your immune system?
I don't think he got it. In those kinds of things like vitamin C is good for the immune system, they're just suggestions. They're not right or wrong. Yeah.
He was not a nutritionist.
No, you don't have to be a nutritionist to be wrong. Nutritionists change their advice regularly. Things like the structure of DNA or the mechanism by which antibodies are formed, you can test them and eventually prove that they're wrong. But whether something's good for you or bad for you is...
So I would request you to go back to your first slide. Can you finally go back to your first slide? First slide.
Who will go back? Not me.
You showed a picture. No, the second one, I'm sorry. Showed a picture there. No, then the previous one. Previous to previous. Sorry, I'm sorry. Previous to this. There is a middle picture you did not explain who that was. That was me. I wanted to bask by the glory of being there in the middle. Professor Talwar, Professor Abdiga, Santosh Kaur. Thank you, Balram. I don't think I can ask question. I'm very excited. I'm very, very excited to have Balram with us. That itself is a impetus for the students, because I've seen him when he was anxiously waiting for getting selected to go to Carnegie Mellon to do his PhD. From IIT Kanpur, he went to Carnegie Mellon to do his PhD. Then he came to Harvard to work with Woodward. Professor Woodward, he worked and then straight came back to Bangalore in 1973, as he mentioned, and he has been there 73 onwards. That is the success story. Not like Santosh Kaur hopping from one place to the other. Yeah, I hopped from one place to the other. I could have been on his tail and become as successful as he has, but life is not just one thing. There are many, many things. I enjoy his friendship. I have not met him for 7 years now. I'm now meeting him. We'll meet more. And thank you, Professor Balram, and give our good wishes to Hema and Aditya.
Thank you, Professor Balram and Professor Santoshkar. I would request you all to come up on the stage for a group picture.
So, can I request you to be the President of the...
Next, we will move on to the Immunocon 2024 award session for the students. I would like to invite Professor G.P. Talwar from Talwar Research Foundation, Dr. Amit Awasthi, President of Indian Immunology Society, and Dr. Kaustav Naik, Secretary of Indian Immunology Society, to host this award session.
So what we'll do is all the students who have spoken, we will be giving awards on behalf of the local organizing committee. The fact of the matter is 6 students have given oral talks and they have been selected out of 359 posters. That itself is a recognition. Right? So we will recognize all students who have spoken, right? The last award will be the G.P. Talwar Award, right? Which the Indian Immunology Society has given. Okay, so can I call upon the first speaker of the oral talks, Babita Negi, to come up? Babita, is Babita around? Oh, yeah, she's around. Yeah, that's a Talwar. Yeah, can you take a photo? The second student is Priyanshi Gogoi. Yeah, the next student should be a little bit ready so that we can do it quickly. The third one is Archana Nair. Yeah. Hold on. Let her finish. Priyanshi. Priyanshi. Archana. Where's Archana? Yeah, I'll call your name. Oh. Yeah. All right. Okay. So the fourth one is Venkata Vamsi Krishna. Oh, it's Vandana. Vandana Vamsi. Vandana Krishna. Okay, sorry. Vandana Krishna? Yeah, it's over. Then we have Akansha Chauhan. The final oral poster speaker was Suraj Singh. Suraj? Okay. I'll now request Sandeep to come and tell us about the poster awards.
Okay. The 359 posters were evaluated by more than 26 people, and they were evaluated based on the not only just quality but also the presentation during the poster session and also the ability to answer questions. So we have 13 poster awards. 4 were sponsored by FEPS Journal. So this time FEPS Journal came forward to give awards to 4 best posters. And I thank Professor Hamza from La Trobe University, Australia, for facilitating this. Thank you very much. So all awards are equal. There is no first, second, third. So these, I call them randomly. The first award is Jaideep Das, FEPS Poster Award. From IIT Bombay. Yes. Okay. Yeah. Second awardee is Swarnadeep Singh from AIMS Delhi. Swarnadeep Singh. Okay, it's not there. Pranit Wahi, NCCS. Yeah. The fourth award is Tanya Sharma from ISER Mohali. Tanya, she's there. Tanya. Okay, Swarnadeep, you can. The Next awardee is Jaideep Das from IIT Bombay. Sorry, already. Sorry. Samarth Daripa from University of Burdwan. Next award is Sakshi Jain from IISc Bangalore. Next award is Preeta Rai Chaudhary from Chittaranjan Cancer Institute.
Preeta is not there. And oh, you are the PI. Come. You can take it on behalf of me.
Next award is Yashwant Kalapala from IISc Bangalore. Is Yashwant there? Yeah, okay. Next award is Prerana from JNCSR. The next award is Kshama Tung from NCCS, Naisar Bhuvaneshwar. Abhiraj R, NII Delhi. Nandini Wankar, University of Hyderabad. Nandini.
Can somebody collect it on behalf of Nandini?
Her PI.
PI or friend or colleague? Somebody from there?
You're Nandini? Yep. And the next one to Krishnendu Chakrabarti from IGIB Delhi. Krishnendu Chakrabarti.
Is he there? Can somebody collect it?
I think he's there. Yeah, he's there. Sorry.
Okay, that's fine. No problem.
Okay, that's all. I'd like to thank all the reviewers who went through each and every poster multiple times and evaluated them according to their quality and the presentation. I also like to thank Professor Talwar for standing here and giving the awards. Thank you very much.
I'd like to now request Kaustuv to declare the GPT.
Okay, so out of the two contestants, I call upon the runner-up first, Rashmi. Can you come out? Yes. The runner-up. And the winner of 2024 Dr. G.P. Talwar Young Scientist Award is Asha Bharadwaj from the Department of Biotechnology, All India Institute of Medical Sciences.
All the students.
Heartiest congratulations to everyone for their awards. I would like to call everyone on the stage for a group picture.
All the students who have got the awardees.
All the student awardees on the stage for a group picture. The American Society of Microbiology has sponsored t-shirts for the winners. I would request all the awardees to collect their ASM t-shirts from the volunteers at the back of the auditorium. And the FIPS poster awardees are also requested to contact our volunteers regarding their prize money for the award.
One award which is missing is for the organizer of this event.
Next, we have a recorded plenary talk as Dr. Jyoti Bajpai from Apollo Hospital could not be physically present. I would request all the awardees to please collect your ASM t-shirts and the FIPS poster awardees to collect their prize money from the backstage. Please contact our volunteers. Come to the right side of the stage, please.
Us to have Dr. Jyoti Bajpai.
We would take a few minutes to set up for the next talk.
is a well-known oncologist working in Apollo Hospital, Navi Mumbai. And she has a dedication to advancing cancer care through innovations and collaborations. She has expertise in various facets of oncology due to her training from leading institutions in India and abroad, including the All India Institute of Medical Sciences, the Memorial Sloan Kettering Cancer Institute, Johns Hopkins University, and so on. Dr. Bajpayee has contributed significantly to the field with numerous publications and involvement in multinational clinical trials. She's played a pivotal role in starting the Indian Immuno-Oncology Society in India and Women in Oncology. In fact, I met her at one of the meetings of the Indian Immunology Society of India, and her enthusiasm was infectious. It may be useful for students to hear directly from her, and so I requested her to deliver a motivational lecture. All of us greatly appreciate that she agreed to deliver this talk despite her busy schedule. Thank you, Dr. Bajpayee. Please start your presentation.
So thank you very much and for this kind words of introductions. And it's a real pleasure and privilege to share this stage. And I'm extremely thrilled that, we are really sharing this passion of advancing immunology and immuno-oncology in the country and, towards the globe as well. And I'm very happy to interact with students during the talk and even later after this. So in the next few minutes, we shall be talking about very general overview of immunotherapy, how it is working in cancer care per se, because I understand that majority of the audience is actually the hardcore scientists, and you guys are doing great work, and bench to bedside conversion, wherein we all are collaborating. So, and what are the main challenges? A little bit about Immuno-Oncology Society. So I really like movies, and a couple of you must have seen this movie, which is The Elephant Man. And in this movie, it is essentially an analogy that how challenging it is when there is some rare and difficult disease like cancer or something else come across. So in this particular movie, which was a critical and commercial success with 8 Academy Awards nomination, forever imprisoned inside a hideous, deformed body, the unfortunate Victorian fairground attraction exhibit, John Merrick, who milked for every penny by the sadistic showman Mr. Whites. And there was many dramatic turns, and later he met a philanthropist who actually helped him, and then the story moves on. So with this, I would begin and I would end with this the same story, what happened at the end, because I really relate the cancer care and especially the advanced, the difficult to treat cancer care with this kind of movie, which is again pose a very big challenge. So we will be discussing very brief about the milestones, the melanoma, the poster child for immunotherapy, because in my limited lifespan, I've seen while I was a resident, and that time the melanoma, the metastatic melanoma used to be a death sentence practically for a
invite Professor Sandeep Ishwarappa on the stage for his talk. I invite Professor Anjali Karande to chair this session. I'll ask somebody.
You could have invited him for the talk.
No, I couldn't find. Yeah,
So good afternoon, everyone. So we've come to the last talk for today, not only today, but also Immunocon 2024. And this is going to be given by Professor Sandeep Ishwarappa. Just a few lines introducing him. So Dr. Sandeep Ishwarappa is a clinician, was trained to be a clinician. He's got his MBBS from the Mysore University, soon after which he joined the Department of Microbiology and ISC to come do his PhD. He went abroad for his postdoc. He went to the US, Cleveland Foundation, I think, Cleveland Clinic, came back to Department of Biochemistry as faculty, where he's been since 2017. Dr. Ishwarappa has been working on genetic manipulation. I don't know whether he's going to speak on that or he's going to touch upon that, but he's recently, and I'd like to tell all of you, he's recently received the prestigious Young Scientist Award, which is conferred by the state of Karnataka, the government of Karnataka. So he's got the C.V. Raman Young Scientist Award, and I think that's really most recent, and we should really. So Dr. Ishwarappa works on genetic manipulation, very interesting, and I hope someday you all will get the opportunity to hear him. But today he's going to speak on evolution and medicine. So Dr. Ishwarappa.
Thank you, Professor Anjali. I know this is the last talk, a talk on evolution in an immunology conference. And just before lunch, already delayed, I didn't expect this many people. Thank you very much for staying back. I try to make sure that my presentation is worth your time. So today, this is not my. Yes, this is mine. Okay. So this is a general talk. It is not based on my lab research. I'm sure all of you know this famous statement that nothing in biology makes sense except in the light of evolution. At the end of the talk, I'm going to convince you that even for medicine, even in medicine, nothing makes sense except in the light of evolution. So these are three photos of three Darwins. Charles Darwin, everyone knows. Robert Darwin is father. Erasmus Darwin is grandfather. Both father and grandfather were doctors, and Charles Darwin was a medical school dropout. So what I'm going to tell you today about medicine is from this guy who dropped out of medical school. So this field of evolutionary medicine was popularized by these two gentlemen, Randolph Ness, he is a psychiatrist, and G.C. Williams, he was an evolutionary biologist. And they wrote an excellent book called Evolution and Healing. I strongly recommend all of you to read. It's a very good reading. So before going to the details, let me explain what is not evolutionary medicine. It is not an alternative to modern medical practice. It is not like other allopathy or homeopathy or Ayurveda. And it does not advocate that medicine should assist natural selection, or it doesn't give any moral guidelines how a doctor should practice medicine. Rather, it is just an additional perspective to look at the mechanism of human disease, and which has been largely neglected in medical profession. As of now, no medical school teaches evolutionary biology as a separate subject, which is really unfortunate. So understand this field, we have to differentiate these two questions, how and why. The how question gives proximate explanation. The biochemists, molecular biologists, and geneticists usually address this proximate explanation, which answers how question, how a disease happens. But why question is more complicated, philosophical, and it gives a broader view. It gives an evolutionary explanation. I'm going to explain this with an example, a simple example, very relevant to this conference, that is fever. If someone asks, why do we get fever? So if you ask a student, the immunology student, it's a very easy answer. There is infection or inflammation, they activate immune cells, they secrete cytokines, they go to a hypothalamus, reset the thermostat, you get fever. But this answer is correct, but this answers how do we get fever, not why do we get fever. So the why do we get fever, that why question is more complicated, more philosophical. So if you expand this question, you can ask why natural selection has favored us to respond with fever for inflammation. Why not something else? That means, is fever advantageous? The corollary of this question, is it correct to block fever? So all of us take paracetamol. We don't go to a doctor. Feeling feverish, go and take paracetamol. Is it correct behavior? So there is lots of tons of evidence which suggests that fever is beneficial. Many animals. Fishes and lizards, they move towards warmer environment whenever they get infected. So people have tried to block the movement of lizards to the warmer environment, and they are more likely die because of infection than those lizards which could move to the warmer environment. And the recent paper, which was published in 2024, early this year, they showed that the amphibian species, which are close to extinction because of this deadly condition called chytridomycosis, it's a fungal infection. So they made these saunas, that is, small chambers which are warmer than the surroundings. They're actually artificially creating a fever-like condition. And these frogs, which are exposed to this higher condition, they could clear this fungal infection. It is not just reduction in infection, it was completely cleared. And they also gained resistance to reinfection. They were resistant when they introduced the fungal pathogen again, they were resistant to reinfection. This is the power of the fever. Even at cellular level, it has been shown that fever aids the immune fight against infection. For example, it allows more white blood cells to enter lymph nodes and fight infection. So this was a paper in Nature Immunology. The power of this fever was realized much earlier. 1920s, Dr. Julius Wagner, he was a physician. He used to cure syphilis by inoculating malarial parasite, which used to induce fever. So remember, this is the era before antibiotics. Those times, an infection could be a death sentence. At that time, he showed that we can clear infection of like syphilis by inoculating malaria. It was such a phenomenal success that he got Nobel Prize in 1927. So overall, what I'm trying to say is fever is an adaptive host response to infection that has persisted throughout animal kingdom for millions of years. That means it is advantageous, it is beneficial. However, it also has complications. It's a double-edged sword. Uncontrolled high fever can cause seizures, delirium, tissue damage, dehydration, male sterility, so many things. That's the reason why, in a high fever, it is beneficial to treat. But in mild fever, it's good for us not to treat and feel some inconvenience of the fever. But still, we can, with fever, we can fight the infection better. Okay, in the next slides, I'm going to show some applications derived from this evolutionary analysis. So we have, the animals have different ways of breathing. These roundworms breathe through skin, insects breathe through trachea, fishes breathe through gills, and many land animals breathe through lungs. But there are some unique and very strange examples. For example, this turtle breathes through the rectum. It's called butt breathing. So these are the loach fish. This also breathes through their distal intestine, which is again through the rectal canal. So it can survive in outside water longer than other regular fishes. This strange breathing pattern inspired some Japanese scientists at the time of COVID. So in COVID, there were many patients who had respiratory failure. They needed ventilator till the respiratory failure is subsided, but ventilators were not available. So that's when they got inspired by these loach fishes, and they thought, why not oxygenate these patients using the rectum? Okay, of course, they couldn't directly go to the patients. They started with some animals. And by the way, this work got Ig Nobel Prize of this year, 2024. So what they did, they took a fluid called PFD, and it is a biocompatible fluid, and they saturated this fluid with oxygen, highly saturated oxygen, and then they gave like enema in these animals, these mice which had a artificially induced respiratory failure. So then they analyzed how they behave with or without that enema. To their surprise, they found they could increase the partial pressure of oxygen in the oxygen saturation in their blood, and also their activity was much improved in the hypoxia chamber when they were given with this EVA therapy. They call EVA therapy. EVA stands for enterial ventilation. So next, these people have started a company called EVA Therapeutics. They proposed to use a distal gut as an accessory breathing organ in respiratory failure patients. If this is successful, many patients can survive even without ventilators. Okay, so the next story I'm going to talk about is about the novel disease of this century, that is metabolic syndrome, obesity, hypertension, insulin resistance, and hyperlipidemia. So the why question here is, why are we susceptible to this condition in the modern environment, right? So if you analyze the root cause, it boils down to this one craving we have, our craving to eat fatty junk food even when we are full. So almost all of us eat more than necessary and more frequently than it's necessary, 4 to 5 times a day. So why do we have that craving? And we know that it is not good for our health, but still many of us cannot control that craving. That's because of our ancestors who were there in African savannas. They were hunter-gatherers. So depending on the weather, the availability of food was unpredictable, infrequent, right? So whenever the food was available, the tendency was to eat as much as possible and store as body fat, right? And only those people who could eat as much as possible and store excess calories as body fat survived, and we are their descendants. So we can't blame ourselves, we have to blame our ancestors for our craving. So this is a nice hypothesis, but another fish in my story, which provides some insights into this hypothesis, some evidence support this hypothesis. This fish, our convener likes fish, so I have 3 fishes in my talk. This is the second fish. So this fish is a deep cave fish. In the deep cave, this is found in Mexico. There is absolutely no light. So over the years, they have lost eyes. Also, they don't have frequent food because no light, that means there is no photosynthesis, no organic matter. They completely rely on floodings. When there is flood, from surface, the water enters the caves and takes all the organic matter. And they eat only that. So in that way, they are like our hunter-gatherer ancestors. They are evolved to live in infrequent and unpredictable food availability. So, of course, some physicians were interested to know its metabolism. They took these cave fishes and compared the metabolism with the related surface fishes. These 3 are cave fishes, Tinaja, Pacon, and Molino, and this is the related surface fish. First, what they did, they measured postprandial blood glucose level, which is normally done by doctors. You can see that all the cave fishes had higher level of blood glucose. This is after a food, so this is postprandial blood glucose levels. Of course, a doctor will suspect diabetes. The next test is glucose tolerance test. So they injected glucose and then measure how do these fishes tolerate this glucose level. The surface fish, it was a very good tolerance. It came down within hours. But in the cave fishes, you can see there was a delay in the decrease in the glucose level, right? So this is a typical impaired glucose tolerance test. Any doctor will say that these fish have diabetes, but they're not. They're just in a wrong place. So the next, these authors sequenced the genome of this fish. They found, surprisingly, a mutation in insulin receptor. This mutation reduces the binding of insulin to insulin receptor. It doesn't abolish, just reduces. So the response of these cells to the insulin is reduced, much reduced. So this makes sure that the blood glucose level is maintained at higher level, even when the food is not available. So this is their adaptation to their lifestyle in the deep caves. And interestingly, the same mutation is found in a monogenic diabetes condition in humans, right? So the nature has already evolved this condition in these cave fishes to take care of the infrequent food supply. So this is the paper which was published in 2018. And what we can learn from here is that diabetes is not a disease. It is just a maladaptation. So what will happen if you take the cave fishes into the surface? They will show the features of diabetes. Similarly, our physiology is meant for such lifestyles, like hunter-gatherer lifestyle. But we are in the modern lifestyles, and this is why we are more prone for hyperlipidemia, insulin resistance, etc. Okay. Third fish. So this is another fish, this not related to diabetes. These are anglerfish. They also dwell in the deep oceans where there is no light and very less organic matter. They're okay with the food, but they have a problem in finding mate. So if there is a female or a male, there is a very rare chance that they'll encounter another potential mate. So, but if they do, there will be a complete transplantation of male to female. This is called sexual parasitism. That means male gets physically integrated into the female. Now, male acts as just a sperm bank, donating sperm to the female. Female, by its vasculature, provides food to the male. So this sexual parasitism is very interesting because this is a case of natural organ transplantation. How do these fish have achieved such a wonderful organ transplantation without any rejection problem, right? And of course, immunologists will be very interested in this. And this paper was published in Science 2020. And what they found, these are the different fish, angler fishes. These are the fishes which show this sexual parasitism because they are deep inside the cave. Some males can attach multiple females. Sorry, some females can attach multiple males. Some females attach only one male. Some attachments are reversible. So whenever there is not required, the males get detaches. But if you see these permanent attached conditions, so these fishes, that the genes which are responsible for T cell and B cell developments are completely absent. That means they have completely killed their adaptive immune system. This is what exactly we do after organ transplantation. We give drugs to completely suppress the immunity, right? So the nature has done already millions of years ago the same thing, but by using genetics, not pharmacological agents. Okay. Last example, there is no fish here, but there is a bat. So this is a vampire bat. This bat is the only obligate sanguivorous animal among all the tetrapods. Means this eats only blood, nothing else. And this is just tiny, like AA battery, 30 grams. Because blood is poor in nutrients, because 75% of blood is water, right? So it has to eat or drink a lot of blood to meet its nutrition demand. So it takes an estimated 800 times more iron in their diet than an average human. Human is about 70 to 75 grams. This is 30 kgs. This is 30 grams. 800 times more iron. So the question is, how is it managing this iron? This is important because we have human conditions where iron excess is found, like hemochromatosis, primary or secondary, due to repeated transfusion or because of some certain mutations. These diseases are common. They are treated with desferoxamine, which is a iron chelator. But can we learn something from these bats, how to treat such conditions? So Some scientists were interested in this question. They sequenced the genome of this bat compared with close related bats. So there were many deletions or complete absence of the genes which were relevant for its flight and their the cooperative, mutual cooperative behavior. But for iron metabolism, the most interesting gene is Rep15. What does it do? So in intestinal epithelial cells, these transferrin receptors take up iron-loaded transferrin and they store the iron in the epithelial cells. And these receptors are recycled back on the surface to get more iron. This RIP15 inhibits that recycling to reduce the iron intake into the intestinal epithelial cells. However, these bats have lost this inhibitor. That means more and more transferrin receptors goes back and brings more and more iron into the cells. So if you see the iron, by staining the iron in the gut epithelial cells of these bats, you see a dense iron overload in the intestinal epithelial cells. So how does it help? These intestinal epithelial cells, even in us, they are very short-lived. They are shredded off every few days. So they're using intestinal epithelial cells as an excretory machinery to get rid of the excess iron. So this can provide a new strategy to treat iron overload conditions. So since my time is up, I will end with this final slide. So the evolutionary perspective of disease not only help us understand the philosophical or the evolutionary reasons for our conditions, but also it provides new insights to develop novel strategies to treat certain diseases. But it doesn't change the goal of medicine. So goal of medicine is to cure sometimes, and to help often, and to console always. Thank you.
Thank you, Sandeep, for that very nice, interesting, and excellent talk. Any questions, anybody?
That's excellent talk, no, for the end of the conference. Now, thing is that you told everything, change of temperature when they go down under the sea. When astronauts fly up, it's called something called space fever. Okay, that's also an evolutionary process. What is important is that all the three model of fish that you have seen, for the immunologist to see the dendritic cells, which is very in the transparent structure of the fish's tails and other things, those can be studied and can be found out what actually causes the thing. And for the vampire bat, one important message is that the Anopheles or other mosquitoes sucks the blood. Okay, there is a leeches that sucks the blood, and that vampire bat. So leeches, lot of what you call anticoagulants and other things. I think those
bats also have
anticoagulants, yes. And this can be a lot of evolutionary finding can be thought about all this process. Thank you for the excellent talk.
Thank you. Thank you. Yes, Sadiq.
Hi, Sandeep. Excellent talk. I just want to relate to very unrelated things. So evolution, when you think of it, is the first thing that comes to my mind is the time scales. It is like over and over years. And now with, say, more globalization, everything becoming more like there are people from different countries now even marrying together. Do you think that evolution has now become faster in terms of just broadly?
Faster. It's very difficult to answer that question, especially when human interference is happening. There is no natural selection, right? There is no survival of fittest anymore. So it is a completely different kinds of evolution. It's an artificial evolution. Natural evolution is going on in its own speed, but artificial evolution definitely changes everything. If you take the dogs, last 100 years, so many different varieties of dogs have come from this size to this size, right? But they all come from the same wolves, the ancestor wolves, which were just a few thousand years ago. Yeah. For the nature to do that, it may take many, maybe thousands of years.
Any more questions? Yeah.
Hi, Professor. Very nice perspective. What I wanted to ask was, especially in the case where you showed the case of anglerfish, absolutely fascinating. So you showed that it has a very depleted, so it seems like it has a very, it's quite immunocompromised in the adaptive section.
In
Does
the
it-
anglerfish, sure.
Yes.
Yes. Yeah.
Does it have a, how does the innate look like? I mean, I see no way how it can survive with that.
No, it has intact innate immune system. And also in the deep sea, we don't find as many pathogens as we find in here. So all the organic matter, life forms, everything is rare. So they just find a way to reproduce by having this permanent sexual parasitism.
But I thought anglerfishes were quite offensive fishes. Wouldn't that mean that they get hurt often?
No. Anglerfishes, you're talking about other anglerfishes which are there near the surface. They don't have this sexual parasitism. Only specific species which dwell in the deep oceans, they have this features.
Thank you.
I want to ask you something little different. Pardon me for that. But basically, I teach immunology, very classical immunology. I don't go into too much of T-cell biology or anything. But the first thing that I try to teach is evolution of the immune system. And the students want to ask why a mammal developed the adaptive and innate immune system, whereas Drosophila stopped at the innate immune system. But then I said it is the way we look at the immune system. What we define in a mammal, this is adaptive and this is innate. In Drosophila, I think they have learned to combine, because even Drosophila makes cells which are phagocytic, which are very much similar to the adaptive immune system. I would next time when I come to Bengaluru, I'd like to come and spend some time with you. I need to your permission. I will write to you about evolution and medicine, evolution and fever, evolution and sleep. Sleep has a very great implication in evolution. I would like to, and the point that you just now made, that evolution as a natural process, an induced evolution, that you can induce the same changes very rapidly. Those are the points that we should teach to our younger people.
Sure. I teach. I take immunology course, though I am not an immunologist.
So, no, I'm not coming to talk to you about immunology.
Sure.
I'm going to talk to you about these aspects, how much of it we should include in our teaching process.
Yes.
So the students start thinking about why Drosophila did not develop the classical adaptive immune system that we understand, but Drosophila survives.
Yeah, see, if you take bacteria, they have an adaptive immune system. The CRISPR is an adaptive immune system, right?
Yes.
So it all depends on how you define.
So can I come and see you sometime
Sure,
in
anytime,
January?
sir. Anytime.
When I come to Bengaluru again.
Yes, anytime.
No,
Yeah,
I'm coming.
always, anytime. Sure.
So, all right.
Yeah. So very nice talk. So
last
regarding the fever part of your talk, we have been taking pharmacological interventions regarding fever. So how does that affect the trend of evolution of fever itself over the centuries?
No, see, we are the only species which inhibit fever by taking paracetamol and its analogs, right? So anyway, our evolution process already we have skewed it because we don't select for fit test or anything. It's all, everything is equal. So this blocking fever separately doesn't make any change. It only gives comfort, but it will also kill your inherent ability to control infection at the beginning itself. So we allow the infection to progress because we feel feverish and we block the paracetamol. For mild fever, according to me, it is not required to take paracetamol. Just take rest.
So thank you, Dr. Ishwarappa, for bringing up the rare in the Immunocon conference. And thanks everyone for being here.
Thank you, Dr. Ishwarappa and Professor Kadande. I would like to call everyone on the stage for a group picture. I now request Professor Ambak Rai to come on stage and tell us a little bit about Immunocon 2025.
Thank you. Very good afternoon to everyone. You can understand I have a very tough job to do. Okay. I have a very tough job to do because ISC has organized and Professor Deepankar Nandi has done really excellent job. So it's a tough job for me. So we have started, and currently means all the formalities are being done, like approval from the ministry, etc. So you need not to go into that. So as you know, Prayagraj, it's a sangam of two river and one is mythical. We don't see that. So you will find a lot of things to be there besides the scientific part. And we would try to make it more enriched, more knowledgeable for everyone. And we are hopeful that you will be there to attend that. So these are the few means the expected date of website launching is 1st of December, and the first announcement will be by 10th of December. So we will be having all the emails, etc., and it will be circulated to everyone. I hope you will join there. Thank you.
As we come to the concluding session of Immunocon 2024, we would like to invite Dr. Amit Awasthi, President of Indian Immunology Society, and Dr. Kaustav Nayak, Secretary of Indian Immunology Society, to share their insights and reflections on the conference. Please come on stage.
Just 2 minutes before the conclusion of the meeting. As a customary, you all may be knowing that before the main meeting, there is a preamble, there is a CME, Continuous Medical Education, for all the immunology conferences which have been holding. So initially we tried that we'll host the meeting at Nimans, but then I immediately agreed when Dr. Deepankar said that he wants to hold it at Indian Institute of Science. Then we thought we'll hold a CME at Nimans. As a customary, it is 1 day CME, but then we made it for 2 days. And 2 days we have called speakers from, very senior speakers from all over the country who were ready to come over. We called about 22 speakers and we gave a full free time for them to discuss, interact with the people in a free way, not restricted time and all that. So both the days went from 9 to 6, 6:30 or 7 o'clock, and it was just a break for either tea or for food. It was really enjoyed by, including students. I also enjoyed the talks. And generally, for CME, the Indian Immunology Society gives about 30 or 40,000 rupees. But I request as a special case, because we held it for 2 days, the committee can consider to give it 80,000 because we didn't get funds from other sources. That is a minor thing for either from society or from whatever the things. So in fact, if I have missed anybody to call for the CME and if they were expecting a invite, I'm very sorry for that because it was a too tight a program and within a short time we could manage getting all that done. But the funds from DST, DBT are a bit difficult to get it. ICMR, me being from ICMR, I could not get funds from ICMR. So I hope those who gave a talk, senior people like Nibriti and others who are not there at the moment, a few of the others, I hope they have enjoyed the talk. Nibriti is there, but others are not there. That's what I'm telling. So thanks everybody for accepting the invite and making the program a great success. Thank you. From Nimans, I congratulate you.
Okay, so we are almost there. I don't want to take too much time, but I think I need to sort of thank different people who have played important roles. And I'll try to run through it, but also give an idea about how this has evolved. Okay, so let me start off. So first is Immunocons are a celebration of immunology, but it is also like an annual meeting of people. So a lot of people, as you can see, meet each other, and they sort of look forward to it. So I think that's something we need to appreciate. But all good things will come to an end. So will this Immunocon. So one of the things is, before the Immunocon, we had an incident. The cyclone was there, so it was raining. It was really, you're wondering how to pull it off. I was wondering whether people will attend, or usually there are a lot of dropouts. A lot of people were worried about orange alerts, yellow alerts, and so on. But most people came, which was very nice. So thank you all for coming, and the people that are here right now. You're the ultimate champions, because most people would have left, but you all are still around. So thank you so much. Okay, so lots of people to thank. I'll just start off with, first I'd like to thank IISc, the Indian Immunology Society, for allowing us to hold the meeting here after such a long time. Actually, by Dr. Taruna Madan, who was the president over there at that time, had broached this idea. I also thank Professor Narendra Mehra, who took, very active interest at different stages. He wanted to make sure that things are going well, and sort of and did the monitoring. Of course, IISc and the Department of Biochemistry, because the way things work over here, everything goes through departments. So that's so thanks to all the speakers and chairs. Some of you have come from very far, and I really appreciate your time and your efforts to come here and deliver talks. So it's involved more, a year and more of planning. So what, for such a large meeting, it is impossible for one person to do this. So one of the things that I did was to reach out to a wide range of people. And I think that is important, might be important for Ambak and others who are planning on holding meetings. I'd like to thank co-conveners Uday Ranga, Dr. Shripat Patil, and the local organizing committee, because you need a cohesive team, because otherwise we can't function, right? So I had inputs, as I said, reached out to different people, because there are some areas where I needed more help than others. So one was, our event manager, who are, very helpful with the transport, hospitality, and things like that, and that I got their contact through Amit. I contacted Jagdish, Mittur, as a way of sort of discussing if some things could be done, and he sort of got us in touch with Biotechnica, who's our media partner. So very interesting, how we got the first funding. The first funding came in from a startup. Now, usually startups are struggling for money, but this was the startup. It's a startup based in Hyderabad that gave us the first funding, and it sort of was very encouraging, because we thought that maybe we will be able to raise funds for this. And this again came in through a complicated route, because this was a BC alumni of the department, and this person had helped this startup, and for some reason he thought the alumni was my student. Alumni was somebody else's student, but it didn't matter as long as we got the money. So it was, it's in India, things don't work very in a linear fashion. It's more complicated. So of course, support from student volunteers, they have done a great job. And we have hired domain experts, and some aspects have worked even when, I don't have to tell them. They just know their jobs, and they've done it really well. So of course, support from lab members, and of course, and family, because it's taken quite a toll. And finally, you can do everything, but still there's a bit of luck. So I think till now it's worked out okay, but we are not done yet, because all of you have to go home safely and reach your home. So that's something. So bottom line is, it takes a village to raise a kid. This is the poster. Now Mohan from Aditi Prince did the poster as well as the abstract book. Lots of people again here to thank for their inputs. Lots of people agreed to come, which was good. This is our event managers, and they own a vegetarian Punjabi restaurant, which is very good. And so again, they helped with the hospitality, transport, especially international transport, as well as stay. And this is Biotechnica. This is Shekhar, who visited us, and we've been in touch. And all the YouTubes, everything is being done by them. So again, I don't have to put faculty in charge over there. Put in domain experts. That's the most important thing. And we needed a lot of help with this, especially the website. And that was done by iCart. And then finally, it was loaded on the IIC server digits. But the only thing is that we couldn't change things in the website very quickly. And that is a problem. So I've realized that. So unless you have control on the website, because things keep changing, dates keep changing because of events. And if you are not able to inform everyone about changes, then it becomes a problem. And I think we did have a problem over there. But both of them, made a good website. But I think it needed to be more flexible. Okay, this is the registration kit. I think most of you are sort of got with that. And this is the abstract book. It took a lot of work. Unfortunately, it reached us on 16th evening, so we couldn't really distribute too much of it because things had already started over here. This is the T-shirt that was designed by Nandini, primarily for the LOC members and student volunteers. I think some others also got it. But I liked it because, she and her sister sort of designed it. She decided, to make something new, not just copy. So I think that was, again, something good. This is a memento, and I put Wajid in charge of that, and Wajid delivered really well. I hope most of the speakers and chairs who have got it will appreciate this. So we started off with the flow workshop, and this is, I think, Dr. Krishnamurthy explaining the basics of flow, and all thanks to Keshav and team to take care of, most of the flow part. So again, I did not have to do much over there. And this is serving food. So that's Mr. Singh and his team had got the food. And I think that's Anantika also helping in serving. You have to multitask. Multitasking is important. With regard to transport and accommodation, I sort of split it because the international part was taken by the event managers. The national part was handled by Dr. Ramchandran and Ravindranath and an entire team. And I think the transport people have, really stressful because things keep changing, flights keep changing, and I think they've done a really wonderful job about transport and stay. And this is part of the transport team. I think this was taken yesterday, and they're still smiling, so which is good. And the food, Deb and myself sort of took care of that. Two Bengalis, so you can understand. Regarding office, we really never had an office. So this is Sirisha, and Sirisha has done all the work. So it was n is equal to 1. So all, so many bills, so many things, and it's taken a long time. And entire handling has been done by her. So it's quite amazing, actually. And this is the cultural show. I think everybody recognizes this kid, our Krishna. And I think, again, so Yakshagana is a sort of important part. I thought it was part to expose people to culture of Karnataka. And of course, then we had Rhythmika because local talent, right? And then Madhura, of course, with her Kathak and Kuheli and this thing with regard to the dance. Okay, so thanks to Professor Talwar for awards and gift distribution. 98 years old and present in the auditorium from morning to night. Amazing, outlier. And then, I mean, it's amazing. Lots of lessons, especially for the young. The stage management was done by Amit, Siddharth, and Samay. So multiple sessions. So they were also in charge of sub halls. Samay was specifically in charge of the flowers, because the flowers that you see, the lilies and the orchids, that's our stable. The orchid color purple turned out to be, the color of the meeting. So that also sort of went along with it. Sandeep handled the posters. Again, 359 posters, judging them, selecting 6 talks, and then selecting poster awardees, huge task. All credit to Sandeep and his team for taking care of that. Regarding funding, so we did get some funding from government sources, and registration, but of course, you have to rely on companies now, because otherwise that's a bulk of the funding came from there. Thanks to all of them. And the way it was done is, IISc is in charge of all the funding. And so all the bills, whatever we get, whatever we spend, everything goes through IISc. So it involves a lot of invoices and things like that. So I need to really thank the IISc admin, because they have spent a lot of time and with help. Last moment, we had to get police permissions and approvals for international visitors. It's a lot of work, and they cooperated really well. One of the advantages of being here for quite some time is a lot of people, and all the people, especially the assistant registrars, the deputy registrars, they were all very helpful, and including people, just normal things. It was really amazing. I have to thank the IISc admin for really encouraging and taking this forward. Okay, sorry, I'm going back. Okay, so regarding the local organizing committee, we have had different meetings, and those are all recorded over here. The volunteers have done, a fantastic job as their mentors. So this is our lab. Lot of effort by all lab members. Thank you so much. Amazing, right? And then this is the entire Immunocon student volunteers. You will recognize this. This is on 16th when we had the mixer. So again, a big shout out to all of them, right? Because, they put in a lot of effort, and, we could not do it. As I said, it's impossible for, one person to run a show. You need a team. It needs to be cohesive. Again, thanks to the family also, because without family support, it's a problem. I was just thinking, after being in the lab for all these years and then visiting all these 5-star restaurants and different places, sometimes you wonder what you're doing. So it's, I just, wonder what it is. Learning lessons also. Is it IIS or IISc? Both are correct. One is the Indian Immunology Society, the other is the Indian Institute of Science. So sometimes there's a mix-up on that. Is it a pro forma invoice or is it an invoice? So again, I had to make these, so I was sometimes a little confused. Same thing. Person asked me, oh, this is for an ODC. And I was wondering, ODC, the way I'm familiar with this is ODC is somebody from, like ODC dance, but it turns out to be outdoor dinner catering. Lots like that. GST, is it 5%, 12%, 18%? Anyway, there are some other fish jokes, but I think Sandeep has already discussed that, so I won't get into it. All right, okay. So there have been challenges. I should discuss it because so that people know. Website, as I told you, was a little bit of a challenge because flexibility was an issue. We had a time when international speakers were being rejected, and therefore we had to figure out how to get the proper category and the approvals for that. Last 2 weeks, in the early bird registrations, we received 300. So it was impossible with such huge load to try and manage. And there, we had to stop because we have our limits on, how many people we can handle. We have security issues and things like that. So these were all, some of the issues, and there were some nasty emails and all, and some, unfortunately, you have to deal with a little bit. So the other thing is we kept the registration fees the same as the last time. For many people, it's somewhat difficult. So, I mean, I think we need to think about it, especially with regard to student travel awards, and see if we can, make them more, because I think they are a group that sort of needs to be encouraged, especially keeping the future in mind. That's something we need to really think about. Once again, I thank you all for coming. I hope you've had good experiences and memories, but I've said, we are not done yet, because all of you need to go home and be safe. I think with that, I think thank you again. I think Amit and Kaustubh will say something, and then we will go outside, take a photo, go for lunch, and then just go home and rest. Okay, all right. Thank you so much.
Yeah, so I'll not take much time. So all said and done, I can tell you that Dr. Nandi and team have set a new benchmark. I can tell you because this is my 22nd Immunocon. So I've been attending Immunocon continuously for the last 22 years. So Nandi ji has set a new benchmark for all, people who are going to conduct or plan to conduct after this. So see you all in Allahabad within in a year's time. Thank you.
Thank you. I will be also very short. So we all echo what Professor Talwar has mentioned, that this is the best Immunocon since the foundation of the society. I echo the emotions, and I think I really thanks Professor Nandi, who put tremendous effort actually organizing such meeting with such inclusivity. This is very important term, because I could see that he accommodated everyone in different roles, right? So this is very important, and that's why the conference become like it's a marathon, because it's a 4-day. I'm staying here 5 days, so, and I keep talking to many people, and everybody is of the same opinion that how such a large conference has been organized such a smoothly. So that means he has put a lot of effort, and I can imagine, because I had organized some conferences, and I understand the pain, and obviously some nasty email we all have to handle. That's okay. But ultimately, this is a great grand success. So hats off to Professor Nandi and his vibrant team. Because as a leader, we can only say that this needs to be done this way, this needs to be done this way. But ultimately, the people who does the groundwork, right? The taxi size time se pahuncha ke nahi, right? So the pickup has happened or not, the rooms were allotted or not, something like even last moment you will get a request ki, sir, mujhe room chahiye, right? Now he has to be on the call, right? So thank you so much. And you have definitely set a new benchmark for the Immunocon with inclusivity. This is very important. We have to include everybody from the senior to junior in some capacity. So this is not only the science. Science is the one part. This is a social event also, where all immunologists from India and outside of India, because some of our colleagues are settled outside of India who has been done PhD with us, they also like to interact with us. So this is a kind of larger family. Thank you very much. And thanks Professor GP Talwar, who was here all the time. That gives us an encouragement that, we also need to listen some of these talks. It's very important. And my evergreen sir, who is here, Professor, you want to stand up? I want to give us some. So Professor Carr is our evergreen, vibrant Now with the Professor Kar and Professor Sarad Dalai, I think society will do wonders, because the guidance from these two senior leaders will help us to go through some of the things which we are thinking to do. And I am looking forward for the next conference in Allahabad. And subsequently, Professor Dil, who is going to organize a conference in Srinagar. So I think we will also, come there. And I also request everybody to become member if you are not a member of IIS, because the society is growing very fast, and I can see it's really a leap, right? And then people like you, if youngsters comes and make this more vibrant and gives an idea. I remember somebody was really in the auditorium saying that, can we have immunothon, right? And this gentleman, I met him again, he's from IISc, and I said, you sent a proposal to us, and we will be love to have this immunothon. You throw some idea and make a team of the students. I remember when Dr. Kanuri Rao was a president, in the meeting of Chennai in 2004, Professor Kaliraj has organized. That time, Professor Kanuri Rao has started this magazine for Immunology Society. And I remember I had an opportunity myself, Dr. Deepaman Ganguly and Dr. Harini, she is now in UK. We started the first edition. So that connection you make here, and this connection will go lifelong, right? So with this, I think I thanks everybody, all seniors, and especially my previous EC, Dr. Taruna Madan, and the team, who actually recognized the importance of ISC to have Immunocon, because I remember the last time it happened here in 1993, if I'm not wrong. So this was really wonderful place to come again. And hopefully we'll keep meeting in the satellite meeting. We are thinking to organize some satellite meeting, more focused meeting from the IIS. And as I mentioned, we would like to go to the village level. We would like to go to the level. I mean, we like to celebrate women's immunologists in India. And we will be shortly sharing you the things what we want to do. And I was talking to Costa, we're going to have 2 years, but we're going to have 2 years sleepless night. I promise we will do everything. And we will do everything to make sure that immunology reaches to every places, including the places where we don't like to go. We like to go to Bombay, Delhi, Bangalore. We like to go to the places from where we all came. I came from a very small town, and many of you came from the very small town. So we need to go back to that place and bring more people, and that's how we can evolve. And with this, thank you very, very much, and we look forward to working with you.
Thank you everyone for coming. This is Deepthi and myself, Shreyoshi, from Professor Dipankananda's lab. And as a part of the student organizing committee, it has been a pleasure hosting you all. We have now arrived at the end of this conference. I request all the delegates to please gather outside on the hall for a group picture in front of the J.D. Tata statue. We will then move on for lunch to the main guest house. Our ushers will guide you to the guest house. Thank you so much for being a part of this conference.